Morning, everyone. It's 9:02, so we're going to go ahead and get started. My name is Lily Mulugita. I'm the Associate Director for Policy and Research in the Division of Pediatrics and Maternal Health at the FDA. On behalf of DPMH and the FDA, I would like to welcome you to our 8th public workshop in the Advancing the Development of Pediatric Therapeutics series. This workshop is planned in collaboration with University of Maryland Center for Regulatory Science and Innovation, and will focus on drug dosing in pediatric patients with renal impairment. I will be co-chairing this workshop with my colleague, Dr. Bakri Al-Zarqa from the University of Maryland. The objective of this workshop is to discuss current approaches to classifying renal impairment in the pediatric population, identify data gaps, and explore scientifically supported approaches and methods for providing information on drug dosing in pediatric patients with renal impairment. So in an attempt to maximize time for discussion, we will not spend much time introducing speakers and panelists. Their bios are available to you online. And a kind reminder also to our speakers to stay within the time allocated to your presentations. We will have someone flagging you as you approach the end of your time, after which you'll have about 3 minutes to wrap up your presentations. In addition, speakers will take clarifying questions during the Q&A and panel discussion sections. So we would really like for this workshop to be an open dialogue among all stakeholders. So we really encourage our audience, both in person. We are expecting over 500 attendees online, so we really encourage the audience to engage, ask questions, and make comments. So without any further delay, I would like to invite Dr. Peter Stein, who's the Director of the Office of New Drugs and CDER FDA, to provide the introductory remarks. Dr. Stein.
Lily, thanks. And in that spirit, I will try to avoid the cane and be brief in my comments as well. Well, first of all, welcome. It's really a great pleasure to introduce this workshop, this very, very important two-day discussion. And I welcome you to this. This is the 8th annual Advancing the Development of Pediatric Therapeutics workshop, or for short, ADEPT workshop. The goal of the annual ADEPT workshop is to gather important stakeholders together to discuss an important topic relevant to the development of safe and effective therapies for children. This year's workshop is no different and is focused on developing a better understanding of drug dosing in pediatric patients with renal impairment. Renal impairment can affect children from birth to adulthood. Regardless of age, the pharmacokinetics of many drugs may be altered by renal impairment and can require the need for dose adjustments. Labeling for most such drugs have recommendations for dose adjustments for adults with renal impairment, but information to support recommendations on dose adjustments in children with renal impairment is usually lacking. Firstly, basic differences between estimation of glomerular filtration rate in adults and children must be recognized. An appropriate method for estimation of GFR in children of all ages from birth to adulthood is a fundamental requirement to provide accurate dose adjustments in pediatric patients with renal impairment. Historically, pediatric patients with impaired renal function have often been excluded from clinical trials. Unfortunately, this exclusion, often without a strong scientific rationale, has also contributed to this lack of information. We also know that, fortunately, the prevalence of renal impairment in children is low, and a discussion of the types and amount of existing data in adults that can be leveraged to help inform dosing in pediatric patients with impaired renal function would be useful. We have made significant progress over the last 2 decades in advancing the development of safe and effective medicines for children. In fact, the FDA has approved over 1,000 labeling changes with pediatric-specific information since 1998. This progress is in large part due to collaborative efforts of the scientific community, clinicians, industry, patient and advocacy groups, and regulatory authorities. In this workshop, we look forward to a robust and collaborative discussion with all stakeholders to develop scientifically sound strategies that it can ensure the safe administration of medicines to pediatric patients with renal impairment. So I welcome you, and I know this will be a very productive and important set of discussions. Lily, back to you. Thank you.
Thank you, Dr. Stein. Our next speaker is Dr. Shamir Tuckman from the Division of Pediatrics and Maternal Health at the FDA.
Thank you, everyone. Hello and good morning. My name is Shamir Tuckman. I'm a senior physician in the Division of Pediatrics and Maternal Health within the Office of New Drugs at the FDA. Over the next 10 minutes, I would like to provide a brief introduction to the Advancing the Development of Pediatric Therapeutics, also known as ADEPT, this being the 8th workshop on drug dosing in pediatric patients with renal impairment. My task in the next 10 minutes is to set the scene for all those attending, both in person and remotely, in what we'll be doing over the next day and a half. Next slide, let's see here. Got it. The views expressed in this presentation are my own and do not constitute an official position of the FDA. In addition, I have no conflicts of interest to disclose. Here's a brief outline for this presentation. First, I plan to provide a high-level overview of why we gathered today and tomorrow, including an overview of some of the challenges associated with establishing dosing for drug products with potential use in pediatric populations with renal impairment. I will introduce the topic areas we'll be discussing at this workshop to address challenges in establishing dosing in pediatric populations with renal impairment. I will briefly review how this workshop is structured. And finally, I'll introduce goals that could be achieved by the end of the workshop. Acronyms. Acronyms are commonly used at the FDA, and the acronyms you'll be hearing in this presentation are shown here. So why are we here? Why is this workshop both timely and important for pediatric patients? Pediatric patients with RI need access to drug products that are safe and effective. Implicit in this need is the establishment of well-characterized dosing when drug product exposures and, by extension, efficacy and safety are significantly affected by changes in renal function as both acute and chronic perturbations. The issue at hand is that current approved labeling for drug products that may be used in pediatric patients with RI does not include dosing for this patient population. To illustrate this point, here's an analysis of drug labeling undertaken by one of our FDA ORISE fellows, Sabrina Rahman. It shows that of 379 new molecular entities approved from January 2014 to May 2022, dosing adjustment for RI was present in 38 labels with an adult indication. An associated pediatric indication was also approved in 9 of these drug products, but only 4 of these labels included dosing for pediatric patients with RI. Of these 4 drug products with dosing adjustments, 2 were antibiotics or antivirals. 1 was a combination drug product meant to treat chronic heart failure, and 1 is a drug product indicated for the treatment of major depressive disorder. None of the RI dosing adjustments came from dedicated RI studies in pediatric patients. Rather, modeling and simulation combined with supportive data from clinical trials, where subsets of patients with RI were included among the general pediatric study population, was used to establish dosing. I will note that most of the pediatric patients with RI from the clinical trials had an estimated GFR above 60 milliliters per minute per 1.73 meters squared. So what are some of the potential challenges that need to be overcome to be able to establish dosing for drug products with potential use in pediatric patients with RI? First and foremost is developing a common understanding of the term renal impairment. Renal impairment in the context of drug dosing could be defined as a change in kidney function that alters the PK and or PD of a drug. Kidney function, of course, can be a broad term that includes glomerular filtration, tubular transport, and metabolism of drug or its pharmacologically active metabolites. In addition, changes in kidney function can be both acute and chronic in nature. For the purposes of this workshop, our discussions will largely focus on addressing dosing challenges in pediatric populations with chronic kidney disease. However, it must be acknowledged that acute kidney injury has important implications on drug disposition and dosing and adds an additional layers of complexity in establishing dosing in pediatric patients who develop acute RI. Another important challenge for all stakeholders, not the least of which includes clinicians at the bedside, is how to estimate renal function across the pediatric age spectrum, accounting for new biomarkers, changes in biomarker assays, and continuously improving estimating equations. Children are in a state of perpetual growth and maturation, and the same holds true for the maturation of GFR and tubular function. How to account for the expected maturation in GFR, as well as tubular transporters that may play a critical role in renal drug handling, is an important consideration for the youngest patients. Clinical trials may often exclude pediatric patients with RI from participation, either because the drug product may have a deleterious effect on renal function, exposure is largely governed by renal clearance of the drug, or concerns that pediatric patients with RI represent a particularly vulnerable patient population to study. Finally, although dedicated RI studies may be conducted in adult subjects, there are very few, if any, dedicated renal impairment trials in pediatric patients for drug products not intended to treat renal disease. This then leaves regulators and drug developers with the limitation of establishing drug dosing in pediatric patients from adult data, which may introduce important limitations. Over the next day and a half, we will work together to try to understand and then strategize or overcome some of these challenges. This workshop is bringing together academicians, clinicians, representatives, regulators, nonprofit organizations, and patients and their representatives, as well as caregivers, to help bring about solutions. Over the next day and a half, the workshop will address 3 focused topic areas. These will be explored through short, focused presentations from subject matter experts, followed by moderated Q&A panel discussions. I will briefly introduce the topic sessions for the workshop. Our first topic area of focus will be centered around what constitutes renal impairment in pediatric patients for the purposes of characterizing the pharmacokinetics and dosing for a drug. Important components that are germane to the discussion of this overarching topic include the strength and limitations of existing estimating methods for specific populations of patients with RI, what clinicians and other stakeholders need to know about these specific pediatric populations, and with a focus on these specific pediatric patients, how should we approach the assessment of renal function within the scope of clinical trials enrolling these patients? Our second topic area of focus will explore how to translate adult pharmacological renal impairment data to help establish dosing in pediatric patients with RI. This includes how, and perhaps more importantly, to what extent, PK data in adult renal impairment populations can be applied to pediatric populations with RI. Is it more appropriate to use body surface area index GFR estimations versus individualized GFR estimates to establish adult renal impairment dosing? And what are the implications with each approach for then using this data to model dosing in pediatric patients with RI? Finally, to what extent can the exposure-response relationship for efficacy and safety be extrapolated from adults with RI to pediatric populations? Our last topic area of focus will be to explore future directions that can be taken to address dosing in pediatric patients with RI. Presenters will put forth illustrative examples that highlight particular challenges and proffer potential approaches to overcome these. We will explore the role of modeling and simulation as a strategy to establish dosing, and when this may be appropriate to use, and what may be the drawbacks and pitfalls of using this approach. Finally, in situations where a modeling and simulation approach may not be appropriate, what are the opportunities for generating clinical data to assess the impact of RI on PK in pediatric patients? So at the end of this workshop, what do I think can be accomplished? Realizing that some of these goals may be aspirational in nature, but potentially achievable. Ideally, we will come out of the workshop with a shared understanding of the key challenges and knowledge gaps, limiting the establishment of accurate dosing for drug product in pediatric populations with RI. We will generate potential strategies that we can use moving forward to overcome these challenges, and finally, be able to articulate common future directions of focus as a shared commitment from the assembled stakeholders. I have included a slide of relevant published studies and guidance documents that may inform some of our discussions today. And with that, I would like to thank you all for your attendance and attention. Thank you very much.
Thank you, Dr. Tuckman. Our next speaker is Dr. Martina Sahre from the Office of Clinical Pharmacology at the FDA.
Hello, everyone. I hope you can hear me better now. So my presentation really is intended to set the stage a little bit in terms of what we do in adults before we then dive into completely the pediatric realm. My disclaimer is the same as Dr. Tuckman's. All of these views are my own and should not be construed to be FDA policies. So renal drug elimination or the kidneys are an important part of the elimination of drugs together with the liver, but we're talking about the kidneys today. And on their route through the body, they encounter, or through the kidneys, they encounter glomerular filtration, tubular secretion, and tubular reabsorption. Those are usually the parts that are considered for renal clearance. And usually when you have kidney disease, you could then have resulting renal impairment that can affect the elimination of drugs, both drugs that are predominantly cleared by the kidneys and also drugs that are cleared by the liver. And so what I usually mean by renal impairment is a change in the capacity of the kidneys to eliminate the drug. But renal impairment obviously also has impact on absorption, distribution, and metabolism, and it can also impact the pharmacodynamics of the drug. There is regulatory guidance out there for how to assess the impact of renal impairment on the pharmacokinetics. That guidance goes into when to assess the impact of renal impairment on PK and PD, where that is possible. It's not always possible. Study design considerations, and then considerations on deriving these dosage recommendations. So when does the guidance recommend, or when does the agency recommend to assess the impact of renal impairment on PK? One is when more than 30% of the systemically available parent drug is excreted in urine. That's also true for therapeutic proteins and peptides, but only those that are less than 6 to 9 kilodaltons in size. So roughly, albumin is the cutoff point here. And then we also recommend that it might be important to assess this for drugs that are eliminated by non-renal routes, because it's been observed that in some cases, metabolic and transport pathways could be affected by renal impairment. And this is less related to renal impairment, but drugs that are used in patients receiving renal replacement therapy, either intermittent or continuous renal replacement therapy, should also be assessed, whether there is a dose adjustment needed. So standalone studies in adults usually, because they are not usually done in pediatric patients, can be designed in two ways. Either you can use a full study design that means that you enroll the full range of renal function, i.e. a normal control group, mild impairment, moderate impairment, and then severe impairment patients, or subjects enrolled into the trial. Although if you encounter somebody in kidney failure, usually they are not really available for these types of PK studies. However, if you encounter them, I wouldn't just necessarily artificially exclude them. And then there is the reduced study design, which is usually the worst case scenario thought for drugs with non-renal elimination. Since the hypothesis is that it is the uremic toxins that cause the increase in exposure and uremic toxins accumulating, the severe renal impairment group here would effectively constitute a worst-case scenario, and so this is why it's suggested to first do the reduced study design in the severe renal impairment group. Guidance recommends that any contemporary equation for estimation is acceptable. That's different from the 2010 guidance where either Cockcroft-Gault equation or the MDRD equation were recommended, and that is really because we recognize that these equations are constantly evolving, and they're changing to some degree in relatively regular intervals. And so we wanted to be forward-looking effectively with that guidance. Another thing to note is that for these renal impairment studies, patients usually are required to have stable renal function or stable renal impairment, i.e. it's not acutely changing. This is something to keep in mind when we're using a lot of the dosage recommendations that are in the labels that are based on chronic stable disease and then having to use them effectively to make dose adjustments in acute disease. Another thing to note is the units. The guidance recommends that the dosage adjustments are based on milliliters per minute. Given that any contemporary equation is acceptable, we do recommend that it's an eGFR that gets used because eGFRs are commonly reported by labs. Those are usually reported in milliliters per minute per 1.73 meters squared. But given that it's the patient's sort of own body surface area that should be considered for drug dosing, the units here are milliliters per minute, and that is also a subject of a talk later today. Data from phase 2 and phase 3 studies can also be used as they often have sparse sample collection, and these can be used for population PK analysis for the covariate effects of interest, and renal impairment certainly would be a covariate or renal function would be a covariate of interest. And when that data is available, you can also use it for analysis of exposure-response relationships, because usually when dose adjusting, we're matching exposures to a reference group. Often that's the normal renal impairment, normal renal function group. But we make an assumption when we do that the exposure-response relationships for safety and efficacy are same or similar, and at least with some exposure-response analysis, you could find out cases where that might not necessarily be the case. One observation that we usually have when we see these study reports is that mild renal impairment and some patients with moderate renal impairment are enrolled, but severe renal impairment is often not enrolled, or there is just not sufficient data available. So how then to translate that to dosage recommendations? Well, to appropriately translate, one thing is to have a good understanding of the relationship between a measure of renal function, that's usually GFR, or eGFR, and a relevant PK parameter, that's usually the area under the plasma concentration time curve, sometimes Cmax clearance or half-life, and then an understanding of the exposure-response relationships, both for safety and efficacy, is also vitally important to take all of the data into account when dose adjusting. And as I mentioned before, dosage recommendations are then based on matching the exposure to a reference group. Like I said, that's often the normal renal function group, but it doesn't necessarily have to be. And the guidance goes a little bit into different means to sort of match these exposures. How does that translate to information in labels for adults? When we looked at approvals from 2014 to 2019, so over a 5-year range, of the 235 approved labels that we considered, drugs that were not considered were some where you just wouldn't have any anticipation that there would be a renal impairment effect, say if they were for ocular use or topical use. And we looked at the information that is included in these labels. So what we looked at was whether there was information in the form of either a statement to adjust the dose, or that a dose adjustment isn't needed, or to avoid use, not recommended, or the use is contraindicated. And no information effectively was either there was no study conducted, or the impact is unknown, or the label was just silent on it. And so what we can see is that for mild and moderate renal impairment, usually there is some information included in these labels. A good 80% of these labels have information. However, that drops precipitously as we get to severe renal impairment and kidney failure. So there is work to be done in adults as well. Switching to pediatrics, again, the two guidances from ClinPharm, again, the renal impairment guidance, as it's commonly called, and then also the general clinical pharmacology considerations for pediatric studies of drugs, including biological products guidance. That guidance gives information on the type of clinical pharmacology information necessary to support safe and effective use. And those guidances, like for the adult case, recommend that any widely accepted and contemporary equation to estimate kidney function is acceptable. However, below 2 years of age, it just states to take maturation into account. And the pediatric clinical pharmacology guidance has some other considerations. Number one, that pediatric patients with impaired renal function should be recruited when it's possible and ethically justifiable to do so, i.e. not by default to exclude these patients. And it also recognizes that data from adults are generally used to complement the information, and that modeling and simulation approaches have a really big impact in this particular area. So how does that translate to labels for pediatrics? We've already heard that today. A paper that came out a couple of years ago looked at lexicon entries that found or that had pediatric renal impairment recommendations. Oh, and I think something got lost here when these slides were put together. So out of the 1,121 drugs, I think it was, that, or one of the entries that they had, they found that roughly 11%, or 126, had information included in these labels or in these entries with pediatric renal impairment information. And when they then went and tried to see whether that information came from FDA label, or not FDA, from the drug label, they found that only of those 126, only 19, or roughly 15%, actually also had information in drug labeling, which left 85% pretty much not having that information. So there is a long way to go to include that information for pediatric patients and labeling. So then why are we here today? So in adults, or as we go through drug development, we usually have data from adults with normal kidney function, and then throughout, hopefully early, because we recommend in the guidance that you should think about that early during drug development. We also then have information in adults with renal impairment, and we often also use that information then, at least from adults with normal kidney function, to predict doses for pediatric patients with normal kidney function, and then test that. But what we're really here for is this lower part here, the part where the pediatric patients with renal impairment, because they usually don't have dedicated renal impairment studies, obviously, and we've just seen that the labeling situation, or that the labeling information is limited in that cohort. And with that, I will. Where's Lily? Yeah, with that, I'm done.
Thank you so much, Martina. Our next speaker is Saskia DeWitt from the Radboud University Medical Center. Thank you, Saskia.
Well, thank you all. It's a great honor to be here from Europe. I'd like to give you a short introduction on drug clearance in pediatric patients with renal impairment, and that was a challenging task to do. So here's my presentation overview, maturation of renal function in kids, the interplay with renal impairment, and what do we know about it in real-life practice. So most of you who have been in the pediatric field for a while have seen this slide before. This is from the 2003 New England Journal of Medicine review written by Greg Kearns about the ontogeny of the processes involved in drug pharmacokinetics, more or less. And it's an old slide, and I keep putting it up because nobody else has made a nicer one, and I can take that as a critique to myself as well. Kids are different from adults, and on the left upper side, you see the change in drug metabolizing enzyme activity, which is low at birth and then increases. On the left, the B You can see the developmental changes in body composition, showing that neonates consist mainly of water, and then this decreases during maturation. On the bottom, the changes in intestinal maturation for transporters, pH, gastric pH, etc. We have filled some gaps over the years since 2003, but I would like to focus on the acquisition on renal function. So this is a very simple simplification. This is a simplification of what happens in the kidney with maturation. And this is, of course, old, but what you can see is that glomerular filtration rate is low at birth, and then increases and reaches an adult renal function around 2 years of age, of course corrected for 1.73 square meters. Para-aminohippuric acid on the bottom, the red line, is represent renal blood flow, but also one of the renal transporters. So when you look at kids, as I showed, and the renal function, it's low at birth, and then the question, of course, arises, so what happens when you have either AKI, acute kidney injury, or chronic kidney injury with respect to your renal function? Does it then decreases by proportion, or do you have a more complicated interplay? As I said, the picture I just showed from the review from Craig Kearns is a simplification over the years, and I won't go into too much detail because Professor Schwartz will go in more detail. But over the years, there has been new data that have been generated using measured GFR, and then trying to come up with better and more precise estimations of the maturation of GFR, especially in neonates, are shown here. On the left-hand side is inulin clearance in a pop PK using population PK, and the different colors represent in the lines represent different gestational ages, and also represent differences between girls and boys, even already in very young neonates. And in the middle is a study we performed with help also from Professor Schwartz, where we took all the measured GFR data we could find in the literature, and we did an individual patient data meta-analysis where we more precisely were able to generate GFR maturation in the first month of age. And we also then related these data with measured data from IOX-SOL to come up with an estimated GFR that can be derived from here instead of the Cockcroft-Gault or one of the earlier Schwartz methods. But what we know about the renal transporter, I just showed you the renal transporter autogeny of one transporter. Work done together with our colleagues from UCSF, Katie Giacomini. We were able to study transporter maturation of different transporters. These are RNA data, unfortunately, from kidney biopsies. And as you can see, there are different maturation patterns for the different transporters. So most are low at birth and then increase, but BCRP, for example, is actually high at birth and then decreases, and some stay stable. But how about the interplay of age and renal insufficiency? How are we going to dose when you have indeed a kid that has renal failure? So in the Netherlands, we have the Pediatric formulary. It's a non-profit paid by the government, similar almost, I think, to Lexicomp, resource where we provide physicians with dosing of drugs, not only the registered drugs, but also the drugs that we use off-label. We are now expanded to cover a population in Europe that's about half the size of the US, with other countries having joined. And in 2016, we received funding from the Dutch Kidney Foundation to actually come up with doses for kids with renal failure. And we had a plan. We were going to do literature searches, and based on the literature searches, we would then come up with doses. But that was a very disappointing exercise, because after we searched about 20 drugs, we just figured there's just not enough data. And then we took the pragmatic approach to then extrapolate from adult data on renal insufficiency, and then provide percentage proportion doses that you could then apply to the already available pediatric doses, not going down less than 3 months of age. So the dosing recommendations are for kids more than 3 months of age. A few years later, we had a PhD student with my colleague, a pediatric nephrologist, Professor Schroeder, and she actually went into the literature trying to actually find out what do we know about all these mechanisms, not only the changes in renal function when you have, of course, renal failure, but also in the other organs, but also if there would be more literature to support dosing. That was, again, very disappointing. It was only a few years later. And again, the advice was to extrapolate from adults because there's not that many data. And here is an overview in more detail. It's from the next review I'll present, where they actually really describe, and for those of you in nephrology, you probably know better than I did, all the differences and all the body organs that change when you have renal failure and how it then affects pharmacokinetics. In our review, we actually looked at what data are available from kids on all these different aspects, so the change in bioavailability, volume of distribution, et cetera, and again, it's very disappointing. Only a very few of these aspects have been studied in kids with renal failure. And again, very recent review in Pediatric Drugs, 2023, where colleagues looked at pharmacokinetics of drugs in critically ill children with AKI, so a slightly different population. They also looked at the data from kids with hemodialysis, peritoneal dialysis, and ECMO. Again, very limited data, but what they did provide, and I think that it is helpful, at least for us in the clinic, is an overview of single drugs, especially how to dose them in these renal replacement modalities and in ECMO, not for kids with AKI in general in the ICU. So how to go from here, and what do we know from pharmacokinetic studies that are out right now, and how can we actually extrapolate? Personally, I'm starting to favor more and more physiology-based pharmacokinetic modeling as a solution to solve this issue. So for those of you not into PPK modeling, and I expect most of you are, it's a combination of existing systems data, such as I just presented for the ontogeny, for example, for kids, the drug metabolism and the renal function, combined with the drug data, so all the properties of the drug, and then you can, with this software, design trials, but also estimate what exposures you will have and how to adjust the dosing. So one of my PhD students, we actually started that NeoDose project for the formulary, is very interested in these kind of studies. And we recently did a search, how many of these PPK papers have been published in Pediatrics, looking at the combination of renal insufficiency and pharmacokinetics. An initial search of PK papers gave us 275 papers, of which 42 are population PK studies. I've not been able to look in detail at all these studies, but I just asked my student, maybe we should have a second student that will look at those data and publish it. But we did look at the PBPK studies. There's 11 that looked specifically at this interaction. There's only very few that actually looked had data to verify these models. And I'll talk more in detail about this approach in my next talk later in this meeting. So very briefly, a little bit disappointing. Maybe you'd hope that I would provide you with real data. Age impacts drug disposition, but the interplay of age and renal insufficiency has not been fully elucidated. There's very little dosing advice, also similar to data that are actually in the label for kids, and also what's on Lexicomp. But the question is, have we been looking close enough at the data that are available? Have we really looked at these POP-PK data that are there and really teased out what is already available? I think there's a little bit more than we all think. I think that we could extrapolate from drugs that have similar excretion pathways, similar transporters, that we might be able to extrapolate, but it takes a bit of time and a bit of work from I think, our community, and indeed the role of PPK studies, I think should be further explored. So Well, I thought it was a very, how do you say, empty start of this meeting, and there's a lot that's coming on how to measure GFR in kids. There's many of these presentations. What is more indeed already known, and what approaches we can take. So thank you. Oh, that was the wrong one.
No problem. Thank you so much, Saskia. Our next speaker is Henrietta Aboud-Kabi from the Office of Clinical Pharmacology at the FDA. As she's making her way here, we have selected one case that exemplifies many of the challenges we encounter as we try to derive dosing in pediatric patients with renal impairment. So hopefully this presentation will provide context both for subsequent presentations, but also for the panel discussion. So, Henrietta.
Hi, good morning, everyone. So I'll be providing a case example on ceftazidime avibactam to illustrate drug dosing in pediatric patients with impaired renal function. Before I get to that, though, this is my disclaimer, which is that the presentation represents my personal views and shouldn't be construed to represent FDA's views or policies. Okay. So for drugs that are primarily renally excreted, impaired renal function typically alters drug PK to the extent that dosage modifications may be needed in adults. And to inform any such need for dosage adjustments in the labeling, PK is evaluated in adult patients with impaired renal function. And it's presumed that renal impairment may also impact the PK of these drugs in pediatric patients. However, for most drugs that are primarily renally excreted, there's a lack of dosage recommendations in pediatric patients with impaired renal function due to the challenges in obtaining PK data in this subpopulation. So with these challenges in mind, the objectives of my talk are first to provide a brief regulatory background on ceftazidime avibactam, and then describe how extrapolation from adult data facilitated dosing recommendations for pediatric patients 2 years of age and older with impaired renal function. So ceftazidime avibactam is a combination product comprised of ceftazidime, which is a beta-lactam antibiotic, and avibactam, beta-lactamase inhibitor. And the combination is approved for the treatment of CUTI, CIAI, and HAP-VAP in adults. Because ceftazidime and avibactam are both primarily excreted and changed in urine, the U.S. prescribing information recommends dosage adjustments in the setting of renal impairment. And for adults, these recommended dosage adjustments are based on creatinine clearance calculated using the Cockcroft-Gall formula as summarized in the table to the right. And currently, ceftazidime avibactam is also approved in pediatric patients 3 months of age and older for the same indications as adults. The approval of these indications in pediatrics is based on the understanding that since the disease course and response to treatment for these infections is sufficiently similar in adults and pediatric patients, the exposures that have been found to be efficacious in adults then would also be expected to be efficacious in pediatric patients. Therefore, the existing adult population PK model was updated with PK data from 3 pediatric studies and used to simulate dosage regimens that would achieve ceftazidime and avibactam plasma systemic exposures, i.e. AUC, similar to the efficacious exposures attained in adults. And Summarized in the table below are the recommended dosage regimens for the general pediatric population, and dosage adjustments are recommended for patients 2 years of age and older with EGFR less than or equal to 50 mLs per minute. However, for patients younger than 2 years of age, there are no recommendations for use in the setting of renal impairment. So then, what are the recommended dosage adjustments in pediatric patients 2 years of age and older with renal impairment, and how were they derived? So the pediatric clinical trials attempted to enroll pediatric patients with renal impairment in order to inform the impact of renal impairment on PK in this population. However, as you can see in the summary table, only 2 patients with EGFR less than or equal to 50 mLs per minute were successfully enrolled in the study, and therefore the effect of renal impairment on PK was largely extrapolated from the adult data. And this was based on the assumption that the effects of renal impairment are proportional in adults and pediatric patients 2 years of age and older, since renal maturation is expected to be complete by that age. However, because the recommended dosage adjustments for renal impairment in adults are based on creatinine clearance determined by the Cocker-Gault equation, and that impedes the renal function was characterized using EGFR, calculated using the bedside source formula. Body surface area normalized EGFR had to first then be bridged to body surface normalized creatinine clearance in order for the adult data to be used to inform or to predict the impact of renal impairment on a PK in pediatric patients 2 years of age and older. So shown on the slide are the simulated ceftazidime AUCs for adults and pediatric patients 2 years of age and older across various, adults and pediatric patients 2 years of age and older with varying degrees of renal impairment. And the exposures are shown both for the recommended dosage adjustments in patients with renal impairment, as well as without the dosage adjustments. The black dashed lines represent the 25th and 75th percentile of AUC in adult patients with normal renal function, and this serves as the efficacy reference. And the red dashed line represents the 75th percentile of AUC in adult patients with mild renal impairment, and this provided the safety reference. So as we can see, in pediatric patients with the estimated normalized creatinine clearance less than or equal to 50 mLs per minute, the simulated ceftazidime exposures exceed the safety threshold, or the established safety threshold. And once the recommended dosage adjustments are implemented, this brings the exposures within the established safety bounds. And so here I have a similar figure for avibactam, and the observations here are consistent with what we have with ceftazidime. And so again, the simulated exposures here for estimated creatinine clearance less or equal to 50 mLs per minute indicate the need for a dosage adjustment. So based on the simulated exposures presented in the previous two slides, then dosage recommendations were provided based on renal function for pediatric patients 2 years of age and older, and the prescribing information. So then, why does the labeling lack dosage recommendations for pediatric patients less than 2 years of age with renal impairment? So in this subpopulation, the rodent equation was used in a pop PK model to describe GFR instead of the bedside Schwartz formula. And this is because when postmenstrual age, or PMA, was used as the covariate for drug clearance instead of eGFR, it provided a better overall model fit. However, in adopting the rodent equation, then the model was unable to account for the impact of changes in eGFR on drug clearance. And ultimately, due to the lack of insufficient PK data to characterize a renal impairment in children less than 2 years of age, there is insufficient information to be able to provide dosage adjustments for this subpopulation of patients with any degree of renal impairment. And that brings me to the end of my talk. Thank you all.
Thank you so much, Henrietta. That concludes the introductory talks, and we're going to move to session 1, which is going to focus on what constitutes as renal impairment in pediatric patients, and this is specifically for the purposes of PK characterization and hence drug dosing. So with that, I would like to invite Dr. George Schwartz to come up for his presentation.
Thank you for inviting me. I've never been to the FDA. This is a pleasure. My goal is at least to give you at least a few translations into some of the abbreviations that were already used, such as GFR and eGFR, and to learn a little bit about what's clearance. I'm not sure we all got that in our training, actually. So from first principles, let's say that glomerular filtration rate, GFR, is our best index of overall kidney function. There is glomerular tubular balance for the most part, and as GFR goes down, many of the other functions are adjusting in a similar way. Obviously, this is affected by very many physiologic and pathologic as well as pharmacologic conditions. The decline in GFR it can be documented very well using both kidney function measurements and estimates. But you can't directly measure the GFR in humans. We can assess it from clearance measurements, and I'll tell you a little bit about that. The gold standard for that is the urine inulin clearance, and this was used back in the 50s and 60s, and now you can't get inulin. So it's difficult to do those studies. It's also it was a difficult assay at the time, and it was expensive, and so it sort of went out of current use. Now we're using more agents like iothalamate, iohexol, and DTPA. I'll point out that there are problems also with these as well. There's probably some secretion of iothalamate, there's some protein binding to DTPA, and a little tiny bit of non-renal clearance of iohexol. So nothing is perfect. Okay, so what is clearance? I had to go back to Homer Smith's original book to give you a good definition. It's a quantitative description of the rate at which the kidney excretes various substances relative to their concentration in plasma. It's a volume of plasma that 1 minute's excretion of urine suffices to remove of urea or creatinine. This is a virtual volume because all the blood is partially cleared. These volumes don't say how they're removed, though. They could be removed by glomerular filtration, as we assume, but there could also be secretion or even partial reabsorption. So the need there is for something that is completely ultrafiltered, gets through the glomerulus, and isn't reabsorbed, isn't secreted, and isn't metabolized by the tubules. Now, inulin did fill this role. Creatinine, as you know, is filtered, but is also some secreted, and some say that the secretion may increase with decreasing glomerular filtration rates. But let's just take this step a little further. We'll assume that our clearance of X is a pretty good marker. So the prime equation is right here. The rate of excretion, which is the urine concentration times the urine flow rate, is equal to the glomerular filtration rate times the concentration of the same agent in plasma or serum. And if we divide by P, we kind of get classic GFR measurement. So it's the urine concentration times the urine flow rate divided by the plasma or serum concentration. That's great, and that's done in adults, and we have this magic number of about 100 mils per minute per body surface area. Now, why do we talk about surface area? Well, Homer Smith back in 1951 suggested that kidney weight would be the best way to normalize kidney function. But that's not something we can easily do, certainly while patients are alive. Now, you could perhaps ultrasound everybody in the world and get some measurements, and this could be done, and the mass could be calculated with certain assumptions. If you can read the ultrasounds correctly, you could come up with a kidney weight or a kidney volume. But an easier way at the time was to use body surface area, and there was a very good correlation between body surface area and kidney weight. And glomerular number, the number of glomeruli in a kidney, which is approximately 1 million, but very variable, is proportional to kidney weight. So now body surface area has been validated as a size comparator, but I'll point out to you that most of the studies that Dubois and others have done in the past had very few children. We've actually gone to a study that was done more recently by Haycock, where many children were investigated, and the body was kind of broken down to the head and cylinders for the arms and legs and trunk, and kind of mathematically added up to calculate surface areas. That seemed to agree pretty well, at least in the adults, with the classical measurements by others. Now, this is a key figure from Homer Smith's book, and I'll point out to you there's a large variation, but what you're seeing here is the age in months against the percentage of adult levels. This is 100% of adult level. These are, I think, 1 sigma, 1 standard deviation. A variety of studies, both premature infant studies done by Henry Barnett, one of the fathers of pediatric nephrology, and others here, that show maturation occurs actually probably by 15 months or so when you correct for body surface area. We were very interested in the adult equations and trying to apply them to children, but we ran into this problem. So as you get bigger, your creatinine goes up, but your kidneys don't get worse. So we had to reconcile this. How do we assume then, as creatinine is increasing with age, and we know kidney function adapts to your body size as it clears the blood of an increasingly large individual, you can't just simply use creatinine. So the adults get away with this in their estimating formulas, but we cannot. And you can also see that we really have to separate boys from girls because the boys do have a higher serum creatinine at a given age. Now, this is probably related to muscle mass, and without being sexist, I'd just like to point that out. It probably is the reason. And we went back to an old paper by Talbot that really looked at this in terms of urine creatinine excretion. These are 24-hour creatinines that are collected in the urine and estimated muscle mass per body weight. And just focus on the last two here. You can see that with emaciation, there is a much smaller percentage of body mass and hence a much smaller amount of creatinine excreted. So we would use this study to think about ways to estimate muscle mass because this might be important as creatinine goes up during the growth phase. And the magic number for Talbot was about 17.8 kilograms per gram of urinary creatinine. So I wanted to just get you to how we estimate kidney function using those assumptions. This is our creatinine clearance equation that basically I gave you earlier, UV over P or S, and the corrected for body surface area. We made some assumptions here. Hope you think they're right. It seems to be okay. Surface area would be proportional to height squared or length squared. In the steady state, urinary creatinine excretion would be equal to the production rate. The production rate of creatinine should be proportional to muscle mass, as I just described from Talbot. So the muscle mass could be proportional to the length cubed. So therefore, the urine creatinine excretion should be proportional to length cubed, and we can plug in everything into the equation, and the lengths go out, most of them, except for one. And that's the guy that hangs in there, so that there's a height over serum creatinine might be a better parameter for us to estimate kidney function. We published a bunch of K values that people used. These were from our original work where the creatinine was measured by a modified Jaffe reaction, a colorimetric assay not specific for creatinine. As you in industry have moved our creatinine assay to an enzymatic assay, we've had to modify things. Now I'll point out that the bedside equation used 0.41, and that was derived from our C-KID study, which I'll tell you about in a minute. But because with kidney insufficiency, puberty is delayed frequently, very few of our kids were post-pubertal. And so we did not see any kind of maturational change in our values for K in our population. This was after 6 years of collecting patients through C-KID. And this study is one of NIH's jewels in the crown, I think, for pediatrics. The study started in 2003. It's a longitudinal study with annual follow-up. 1,100 participants have been enrolled. Over 50 sites in the U.S. and Canada are involved. We focus on progression, cardiovascular comorbidities, growth, neurocognitive development, and a little bit now into genetics. We measure GFR in all our children by plasma iohexol disappearance. We chose plasma clearances because, as you know, about 80% of pediatric renal impairment, I'll call it kidney impairment now as an ASN does, is non-glomerular. Hence, there are going to be bladder as well as emptying issues with many of our children who have chronic kidney disease. Therefore, we thought a plasma disappearance would make more sense than having kids try to urinate, especially 3-year-olds and 2-year-olds who are not toilet trained. So we used iohexol. It has a lot of features that are indicate it's safe, it's non-ionic, there's no protein binding, there are very few side effects unless you're allergic to shellfish, and that's probably the iodine issues. It's not secreted, metabolized, or reabsorbed. There's a very small amount of extrarenal elimination, about 3 to 4%. And this is kind of how we do it. We started off with 9 points, and we reduced them to 4, realizing that this is more of the distribution, and this is then more of the renal phase. And the dose divided by the area under the curve would give us the GFR. In the first 350 children, you can see that there's a significant amount of renal impairment. The average is about 44. And here's the study from 1976 showing height over serum creatinine well correlated with the measured GFR. And this is what we found in our first group of those 350 children with CKID. The same parameter, height over serum creatinine. So we thought this was a pretty good way to mark GFR. And then subsequently, we've done our update with the under 25 equations, where we have actually tailored the K to accommodate both being a male as opposed to a female, and being, we are overestimating our kidney function in the little kids, and that may be why you use that PMA, the Rodin equation. So we've dropped our Ks in the premature, in the under 12 there, and we've extended our data up to 25 with, again, a fairly constant estimate up here. So that's been our modeling. This is all done based on curve fitting, and this makes sense from a point of view of puberty with creatinine. We have no idea about cystatin C, and I'll have to talk about that a little bit if there's time. But this is purely empirical to show that there are changes also for boys and girls with cystatin C. And if you look at the equations, assuming a constant, K, we do have overestimations and underestimations in the older children, and when we use these adjusted Ks, we've reduced the bias to virtually insignificance, both in boys and girls, and similar findings for cystatin C. These are the raw values for iohexol. You can see pretty well correlated by a creatinine-based estimate with those Ks, and this is for cystatin C. I've got the red light. Should I stop, or what would you like me to do? Keep going? Okay. These are a variety of other equations that are out in the world, the full age spectrum from Europe. There's this Lyon equation, there's a few others, and we looked at it in our testing group and found that the bias was obviously not as good as our equations for cystatin C and creatinine. And that's true for both under and over 18 years of age. So we've provided these relatively simple formulas. There's now an app. You can actually get it, and I use it in clinic all the time. And it gives us our GFR. It's a little more accurate than the bedside because it's adjusted for age. Now, I wanted to throw out some things very quickly then. We've looked at the CKID population derived data and applied U25 to a variety of studies that we found that came out. We also validated in normal children, and we looked at some discordant cases very quickly through these. This is from studies in our testing group. We looked at the CKD-EPI and the European Kidney Function Consortium. And you can see for creatinine that, this is bias. This is the percentage of GFRs estimated that are within 10% plus or minus of the measured, and this is 30. And you can see performance is not bad for the EKFC as well as our under 25. CKD-EPI is absolutely not a very useful equation when our kids are involved here. And these, again, our kids are over 18. For cystatin C, our equations seem to do better than EKFC and certainly CKD-EPI with bias and percentage within 10%. But the most accuracy was found with a combined, using both a creatinine and a cystatin C-based estimate. We think that gives you the best accuracy. So the U25 equations that we generated from CKID have outperformed the CKD-EPI and the EKFC in our testing set. I won't go through the details. EKFC was a little better for creatinine, but not for the cystatin C. And then we came across a paper by Nyman, AJKD, looking at normal, basically normal European children, which was a group that he studied for a variety of reasons to justify equations developed in Europe, but they also happen to have the creatinine-based U25 in there, and you can see that it did pretty well as well. The bias, oh, sorry. Is that the right one? No. Very hard to work at this angle, but I wanted to show you the bias is really quite good in older children who are have normal GFRs as well as younger children. So these are the older children, these are the younger children. So pretty good. The cystatin C formulas didn't work quite as well in the European kids. And then if you look at when measured GFR is less than 75, this is the difference between cystatin C and creatinine-based estimates. It's virtually no bias between the two different estimates, and there is a bias in the higher GFR patients there. So U25 creatinine does pretty well in European children and young adults with normal GFRs. Then we just looked at discrepant cases within our own testing set. So for example, if you look at the top here, if the creatinine estimate was more than 30% higher than the cystatin C estimate, you took the average, you're very close to the mean of the measure. The average of these two measurements is very close to the measured GFR. And the same is true if the cystatin C is 30% higher than the creatinine-based estimate. And I'll give one last presentation here. This is a group of about only 29 children who had near normal GFRs, and there was a question of what do we do about them in terms of they have discrepant measurements. So in this case, if the GFRs were equal on estimate, any of the equations worked pretty well, and we tested the bedside and the older CKID equations as well as FAST, which is the full age spectrum European equation. And when creatinine-based estimates were higher than cystatin C estimates by more than 15, the U25 creatinine or the combined would be the best measurement. And you can see here when cystatin C was the higher one, the U25. So it looks like overall, if you get both the creatinine-based and the cystatin C-based estimate, you have a very accurate measure of GFR. It seems to be better than just using creatinine alone. The limitations of any of these estimates is critical illness. The assay, half our PIs didn't know what kind of assay was done at their own hospitals in terms of whether it was a Jaffe or a creatinine-based, enzymatic-based assay. Rapidly changing serum creatinine, you cannot use estimates. Muscle wasting or heavy weight training will certainly change your ability to use creatinine-based estimates. So if you don't have success in knowing what your GFR is, these are things you can do. And we recommend this all the time now to get both. Don't repeat the creatinine, but get the cystatin C and take an average. Or you could do the old-fashioned clearance measurement, collect urine on an older kid who can urinate. With cimetidine-inhibited creatinine clearance, you can see that cimetidine reduces the amount of creatinine secretion. You get very close to the measured GFR. Or if you have it, iohexol or iothalamate. And I'm sorry I ran late. I really apologize.
Thank you, Dr. Schwartz. That was a very complicated topic. We asked him to cover in 15 minutes, so it was definitely understandable. Our next speaker is Guido Filler from Western University.
Thank you very much. I appreciate the opportunity to share some of the own work and some of the work of others. The objectives are really to identify special population where we have to identify special ways of estimating the GFR. I also would like to talk about the renal development a little bit more, especially in view that in downtown Minneapolis we have 16% prematurely born children and worldwide about 11%. So that's a large increase, possibly due to microplastics, and we have to, of course, accommodate that growing proportion of prematurely born children. And I would like to talk a little bit more about the challenges of switching from pediatric to adult formula. So the definition of a special population is really that they are characteristics that render the usual approaches like the U25 unusable. So, for instance, not a steady state, abnormal muscle mass, and conditions that affect the body surface area calculation, change in metabolism, and then if the biomarkers are altered. So the first group that I would really like to focus on are the newborns. You really shouldn't be using creatinine at all for the first 3 days, and ideally not for the first week. Creatinine crosses the placenta, and even at 72 hours, there's a very strong correlation between the maternal and the neonatal creatinine. The amount of cystatin C is minimal, but it's not zero. Also, cystatin C is very heavily expressed in the placenta. And currently, the only biomarker that does not appear to cross the placenta is actually beta trace protein. And another way of looking at neonatal renal function when you don't yet have that hyperfiltration is to look at the renal volume, and looking at renal volume Z-scores. Karen Abbott-Ball from the University of Miami has been working a lot on that. A very interesting approach is to use aminoglycoside levels, or possibly also vancomycin levels that could be used independently for estimation of GFR. But of course, we want not to expose any children to that drug, right? It's only an opportunity when they get the drugs. So here's just an example of how on day 3 you get that strong correlation between the maternal and the neonatal creatinine. Factors that affect the neonatal creatinine can diminish or it can increase the creatinine. So placenta function, maternal hyperfiltration can lower the creatinine in the baby. The infant GFR obviously affects that. Prematurity affects that. Nephron recruitment and non-renal elimination and catabolism. Factors that increases are decreased GFR of the mother, preeclampsia, and then the infant muscle mass obviously also constantly changes the creatinine production, and if there's anabolism. So when we have a newborn with chronic kidney disease from congenital anomalies of the kidneys or urinary tract, the creatinine rises rapidly. It has been beautified by the mother, and then it peaks at a certain value, and then it lowers itself. When you plot the cystatin C eGFR using my old formula from 2003, then there's actually that steady increase in parallel to the recruitment of this. So the take-home message from this is that for neonates, we cannot use creatinine at all, and we should be using cystatin C. So what about infants and toddlers? So we've learned just from Dr. Schwartz that it takes 15 months or 18 months for the GFR to approach the adult values. And we really should be having age-independent Z-scores to calculate the GFR. That is normal. But as you can see, there is obviously a significant improvement of the kidney function with the recruitment of nephrons that actually occurs in the same order as they were generated. And for the drug development, that's very important because you got away with once daily dosing with vancomycin in a newborn, but have they been in the ICU for 4 weeks, then that's inappropriate. And we actually don't really have a good algorithm for that. When you look at cystatin C by age, there's also that adjustment. I had a chance here at Western to look at a large number of normal children and what happens to the cystatin C, and you really need, again, age-independent, you need curves that adjust that. And the interesting thing is that the GFR is a little bit higher than what you get with the creatinine-based methods for the adjustment. This is the attempt to combine all the data. So based on cystatin C. So cystatin C doesn't change in the prematurely born babies. So from 25 weeks onwards, we are now resuscitating even 23-year-old. And the important concept here is that the nephrogenesis almost arrests when the baby are born prematurely. So we're actually having a tsunami of children that are growing up that were born prematurely. If they were born before 36 weeks of age, they will have a reduced nephron endowment. And then it increases, and this is a logarithmic scale, and it's a lot of data that around the birth point, that's why the curve is a little bit jagged. And I have the data, but I don't have the statistical methods to make age-independent Z-scores from this. Another important group is the patient has abnormal legs. If they have abnormal legs, we cannot really properly establish the height, and that makes it difficult to establish this. Now, there are some solutions for that. We published this in Pediatric Nephrology. You can use two things. You can use the sitting or the knee height to calculate this. And here are some formula that I'm not expecting you to memorize in any way, but they actually work very well, and you can overcome that problem. The knee height is basically the distance from the posterior surface of the thigh to the sole of the foot. So if a child is wheelchair-bound, then you can use that, and you have to adjust it by race, which I hate to do because it's really a social construct and not a biological parameter. But Chamblier published this, and that's another way to establish the total height, the total stature. Another group that is very important because according to some national statistics, for instance, in Taiwan, the fastest growing rate of new patients with end-stage kidney disease is in patients with spina bifida. And in spina bifida, creatinine does not work at all. So some of our own work, but has been shown by others, you have a much improved performance if you use cystatin C only. And here is an example where you should not be using the combination with creatinine at all. Spina bifida is most commonly in the lower lumbar spine, and it affects your biggest muscles, the quadriceps and the leg muscles. And if you don't have those muscles developed, the creatinine is not correlated to the height anymore. So it has been validated externally that you have, compared to nuclear medicine scans and others, that you have to use no creatinine in these patients. Another very obvious group is muscular dystrophy, right? Muscular dystrophy patients have very abnormal muscle mass, and they should be also not be studied with creatinine. You can get creatinines of 10 micromoles in a 25-year-old, and the best formula that work in these patients are cystatin C only, Zapitelli, Larson, or my own formula that works with a very good agreement in those patients. Now, what about some other populations? So if you have a very high muscle mass, and we get these patients quite often, adolescents that work out, sometimes they take even creatine supplements, and the creatinines are falsely high. So again, and George showed some of the examples of where you get creatinine, discrepant measurements. Cystatin C may actually be the more accurate. And actually, very strenuous exercise can temporarily decrease the true GFR. Also the animal protein intake, the creatine intake, can affect the creatinine. If you have an excessive protein diet, then you have perhaps a little bit of hyperfiltration, but not a decrease in the GFR. And again, here, a cystatin C only based estimation of GFR may be more appropriate. So for the pharmaceutical industry, and those who want to plan some studies, I think it would be helpful to look right away at the discrepancy between creatinine and cystatin C eGFR and find algorithms to say in which conditions should you only use cystatin C. And then there are unfortunately some groups where nothing works. So oncology patients with the lifetime cancer rates in Ontario with 46%, we have to brace for enormous increases of cancer, childhood cancer as well. And the patients may have had a normal kidney function pre-treatment, but the chemotherapy can affect the renal function. And they're often renally excreted, and they have a narrow paratherapeutic window, so it's very important to assess the GFR properly. But unfortunately, in the studies that were conducted, neither the creatinine nor the cystatin C base may actually measure it accurately, and you have to rely on actually nuclear medicine scans for those patients. We had a chance to study this in cystic fibrosis patients. So the problem is that none of these markers are ideal. The creatinine depends so much on the muscle mass and the nutrition. Cystatin C is affected by inflammatory states. It's affected by hyperthyroidism. It's a protein, so it is still affected by other factors. So we chose a very strictly controlled group of cystic fibrosis patients, so that's why the group is not very big, but they had to have multiple courses of antibiotics and Pseudomonas infections and had some wasting disease. And as you can see, the slide is very, very busy, and I'm not expecting you to everything, but within 10% is not acceptable for any of these patients, and you may have to use nuclear medicine GFR. A group that needs special attention are young adults. It was already alluded to that a discrepancy between CKD-EPI and the PSU25, and you get this very implausible increase of the GFR by 30 milliliters per minute, 1.7 meters square, when you switch at age 18 from pediatric to adult formula. And that's making no sense. And we actually had a lot of patients that were discharged from adult nephrologists after transition because they used CKD-EPI. The first point about it is that CKD-EPI was 3,995 patients, and it was from 3 different continents, 3 different methods, but less than 30 had a GFR of under 30. And you can only use estimations from a regression analysis that had data points, right? We had a chance to look at this at a large group of patients that were followed longitudinally through a transition program. And I'll cut right to the chase. There is no agreement until you reach age 25. Age 30, actually. And there's discussion. Is it 25? Is it 30? Celistra and others have actually said also in the portal for age spectrum, it goes until 40. And we need to look at these young adults and to come up with better ways. I think CKD epi is not the ideal way to look at them. We already showed the discrepancy as another example, and you get an overestimation with CKD epi. So a take-home message is under the age of 25 for sure, but possibly even younger, even older, do not use the CKD epi formula. Another study here by Celistra showed the same thing, and the big advantage of this study from Lyon is that they actually had measured GFR in those patients. We have to use the pediatric formula until a much larger age group. I agree with George that the U25 works really well, but when you analyze special subgroups of that, especially if they have a normal or a hyperfiltration GFR, then the U25 does not work very well. And we had a chance to look at this in 240 patients that had a measured GFR, and it was a very, very strict way of measuring the GFR. In addition, we normalized the weight to an ideal weight and did not use for the calculation of the body surface area the actual weight, because if you're obese, you underestimate the true GFR. If you are very thin, you overestimate the GFR. And as you can see, the portal for age spectrum and the U25, they deviate quite a bit when you have a GFR in the higher range. And we came up with a new way of doing this when we used the modified Schwartz and my old filler formula, and within 10% and within 30% for a GFR of over 75 worked much better. And the agreement in the lower range was actually quite similar to the U25. The challenge of this is that it has not been externally validated. So to summarize the special populations, while serum creatinine-based eGFR is widely used in special population, it may yield very misleading measurements, and they often have comorbidities that require closer attention to the kidney function. And the wrong eGFR for drug dosing has very significant complications. When we look at vancomycin dosing, we did a review with the best available modern algorithm for vancomycin dosing in neonates and in prematurely born babies. And with that, we had only 19% of the vancomycin dosing right in our hospital, despite having used the best. I am dreaming of a website, how do I get my vancomycin dose right.com, and a Bayesian calculator where all the data are entered, and then it continues to grow so that we can improve the safety, because most of the AKIs in prematurely born babies are now because of vancomycin. And special population require alternative methods for accurate kidney function method, and I have given you some examples how to do that. Thank you for your attention. If you want an educational review, here's one paper.
Thank you so much, Guido, for that excellent presentation. So we'll switch gears to the industry perspective, and I'd like to invite Ash Sharma to start us off, and followed by Nicholas Webb and Deepa Chand.
Good morning. So I will talk to you all about the bioanalytical aspects to consider, and also some of the non-GFR aspects to consider when you think about formulas to use in GFR assessment for patients. So this is my conflict of interest. I'm an employee of Boehringer Ingelheim Pharma. The concepts that are presented here, they reflect my opinions, not that of my employer. And some of the concepts presented here are developed and discussed as part of the IQ Consortium. I work in the organ impairment group there. This has also been published a couple years back. So I'll start off here with this diagram, which shows some of the markers and equations for renal function assessment. You have, I think Dr. Schwartz very nicely showed, we have the, how to switch from here. You have the insulin clearance, which can be gold standard for measuring GFR. You have, of course, the alternative methods with iohexol, iothalamate. And we have also the possibility to go with creatinine clearance measurements with urine collection over 24 hours, etc. But what we use most often in our work is, of course, the estimation of GFR using some of the equations. We have the creatinine-based equations, and this list is by no means like a comprehensive list. It just has some of the formulas that are used very often or have been used very often in the development work. We have cystatin-based equations. We have creatinine and cystatin C-based equations. And I think, as my previous speakers have pointed out, these endogenous markers are used quite frequently in our work. So now coming to the analytical aspects for measuring creatinine. Many of you would know the method for measuring creatinine goes back all the way to 1926, and this is from Jaffe, called Jaffe method, and it's simply taking a sample, adding folic acid in an alkaline medium, gives a yellow-orange color, which is measured photometrically, and it hasn't really evolved in that much. This reaction is very unspecific. It also picks proteins, glucose, ascorbic acid, ketone bodies, pyruvates, guanidine, cephalosporins, aminoglycosides, and bilirubin. I think when you see a list here, it's easy to say that there'll be issue if you have patients with diabetes in your trials, or if you have patients with liver problems in your trials. I think besides the original Jaffe method, a lot of changes have come in the method over the years. There have been changes made to try to get rid of some of the unspecificity. There are some empirical changes as well done by some labs to go around these problems with the unspecific reactions. The other newer methods that came were the enzymatic methods. These involve a reaction of an enzyme with creatinine creating a red dye, which is photometrically measured. The interferences that I mentioned above, they are vastly reduced. I think there's still some reaction with dopamine and dobutamine, etc. But most of these interferences that are common, they are not an issue anymore with the enzymatic method. So it would perform much better than Jaffe method in some of the conditions that I mentioned earlier. Now I go to another part about creatinine measurement, which is called the IDMS standardization, so isotope dilution mass spectrometry standardization. There were big concerns about inter-lab variability with different labs using different internal standards. And the National Institute for Standards and Technology released a standard reference material. This is SRM 967A. A lot of the manufacturers, they changed their equipment, they changed their reference standards that they sold to be compliant with this IDMS reference standard. And nowadays, the creatinine bioanalytical methods, which are traceable to this, are called the IDMS traceable methods. The point here that's important is that the output between IDMS traceable and non-traceable methodology can vary up to 20%. And the newer equations that you have seen, and they are intended to be used only with the IDMS traceable creatinine measurement. Going back to this slide here, the enzymatic methods are all IDMS standardized. But asking different labs around the world, we realized not all labs have actually used the IDMS standardized Jaffe method. Some do, some don't. think I'm missing one slide here, something I have missed out. That's about cystatin. Cystatin is nephelometrically measured, and there's also a standardization for cystatin C that has come recently, and that's the IFCC calibration. That also is important for the equations. Now, just looking at the adult equations, this form, of course, basis for our labels and our dose adjustments and exposure analysis, which we take into account when we are going through the pediatric part. The original Cockcroft-Gault equation and the MDRD equation that came out at the beginning, these were using the non-IDMS standardized assay. But all the equations that came afterwards, including the modified MDRD method, the CKD-EPI method based on creatinine and creatine cystatin, these all use the IDMS standardized assay. Also, this applies, of course, to the race-based refit that came out in 2021. The pediatric equations, and here you have, I think the original Schwartz equation was not, but I think everything else that has come out that's based on creatinine, to my understanding, is IDMS standardized. The cystatin C-based equation that came out in 2012 used non-IFCC calibration, but I believe the U25 equation is based on IFCC calibration. Now, this will be an odd slide here where I'm talking about why Cockcroft-Gault formula or Schwartz formula should not be used now. The reason being, I think there are several-fold reasons here. The Cockcroft-Gault formula, as we realized in our survey, it was in fact being used by some companies to look at the GFR, the creatinine clearance for adolescents. This is what we found in our survey. And this we feel should not be used because, first of all, this is using the original Jaffe bioanalytical method, and you would have overestimation of renal function here. Also, it doesn't look into demographics because only white men were used when the equation was made back in the day. And also, based on using the original Schwartz equation, you would also have overestimation because this also uses non-IDMS standardized output. So I think what's important at the end of the day is understanding where your labs are, what the labs are doing. So if your labs are utilizing non-IDMS standardized serum creatinine assay, you have to consider whether you want to use them for your work. And I think if you're working in the U.S. or in the West, this is not an issue. But our trials are worldwide. We have centers all over the world, and we cannot be sure what the labs do. So what we do is try to actually find out what the labs are doing in terms when they take samples for us. So it's important to be aware if the non-IDMS standardized Jaffe was used, or if your non-IFCC standardized cystatin was measured in the labs, in the local labs. And if this is done, we try to note that down in our database and at least try to use an alternate formula than the formulas that we would prefer to use in our work. I think one other thing I want to point out here is I think with creatinine, what we find, what's important is the creatinine based assays are very, they're quite accurate now, and there's a lot of these are inexpensive to apply, whereas the formulas that use cystatin, they are not as common, and the calibration part is also not easy to be sure of. So that's still something that needs to be more standardized and more available around the world for our work. I think one other part is this is based on experience in our work, in our development work. If the drug that is being developed is an OCT or MATE transporter, you would find that your creatinine values may get biased if you're trying to look for your GFR estimates. That's something to be kept in mind in drug development. And also, if a patient in your trial is taking one of these drugs, they may have a different readout compared to other patients. In that case, the statin-based formulas could be more helpful. Then going to some of the factors that can affect the creatinine levels. One is exercise. I think there's been publications that show a huge increase in serum creatinine if the volunteers had done like a rigorous exercising. Also, there's besides exercise, creatinine being something that also comes from cooked meat. This is also the diet also has an impact on what we will measure as serum creatinine. So if you see the serum creatinine values, sorry about that. If you look at the serum creatinine values, or you look at the GFR values for patients who had taken cooked meat, you would see a huge bias in GFR, especially in the ones who the lesser their GFR, more the bias you'll get. In contrast, non-meat-based meal does not bias your GFR estimate. So some of the other factors that can affect GFR estimate would be some of the medications I think I mentioned, cephalosporins and fenofibrate, cimetidine, besidrazine. Some of these, either they are interfering with the assay or they're interfering with the secretion of creatinine. So there are different ways why they can interfere with your GFR estimate. RAISE, I think, and creatinine-based assays are not of much use, and you have cystic fibrosis, kidney transplantation, or pregnancy as some of the conditions. Cystatin is also affected. I think obesity, thyroid disorders, HIV patients with a viral load, cancer, steroid therapy, inflammation, and smoking can impact cystatin levels. So further, at least what in the organ impairment group we feel with all the discussions we had, it would be good to have an indication in the drug label which equation was used during drug development. If we look at some of the older labels, even they use a completely different cutoffs. You know, instead of 90 mils per minute, you have 80 mils per minute, which is a normal renal function. And you don't know if they use Cockcroft Gault, or whether the Cockcroft Gault were used for adults or whether used for adolescents. So they use a different equation for pediatrics. So it will be good to have that shown in the label. And it will be good wherever possible to stay consistent with the equation in the drug development program. One has to be always ready to adapt an alternate formula to adjust the dose of for a drug in a clinical trial if necessary. So that's everything from my side. Thank you.
Thank you so much, Ash. I'm going to ask Deepa and Nick to come up for the next presentation.
Thank you all. And as our disclosures, Nick and I are both employees and shareholders of Novartis, and our views that we're expressing here today represent ours and not necessarily those of Novartis. And what we'd like to do is maybe take a step back, and as we've been talking about pediatric drug dosing, let's talk about pediatric drug development. And in order to do that, we'll talk a little bit about adult drug development and how it pertains to pediatrics. We'll also discuss the renal risks that can be associated not only with the product, but also the underlying state. And when we start developing drugs and as we're establishing dosing, there's a few key principles that we need to keep in mind. We do monitor molecules throughout the entire drug development process, and we're looking for that nephrotoxic potential all along the way. And what we've done internally is developed a schema by which we can somewhat quantify and somewhat stratify that renal risk so that we know how to look at that, and then we can put in place monitoring as well as safeguards for the patients. And what we've done is that we've defined compounds based on certain criteria as either having a low nephrotoxic potential versus having a high nephrotoxic potential. And that's namely based off of our preclinical data that's done through animal models, either in vitro or in vivo. And we look at safety parameters, including GFR measurements, including biomarkers, et cetera. We also look at the mechanism of action, and we see how well characterized it is, and if it's something that we've seen nephrotoxicity in, and have we seen this consistently throughout some of our early development phases. And then we look at what have we seen in clinical studies, and namely in human studies. And as we mentioned, sometimes pediatric data is lacking, but what we do is at least we start with the data that we have and try to determine this on a compound level. And we also have recognized that there may be predisposing conditions to nephrotoxicity. In children with renal impairment, we also need to consider these on top of the compound itself, and some of the factors that can affect that toxicity that could occur. For example, there may be altered pharmacokinetics due to changes in absorption. We've talked about body surface area. We've talked about lean body mass. These certainly all affect absorption, distribution, metabolism, or even the excretion of these compounds. There may be interactions with medications. As Guido mentioned, there are certain special populations that have chronic kidney disease and have chronic alterations even in metabolism. And we have to look at that not only in the context of the patient, but also any other medications that these patients might be taking, which may alter the renal function within itself, either directly or indirectly. We also need to consider the mechanism of action that could affect the kidney, not just on a holistic level, but within what components of the kidneys. We've spoken a lot about glomerular filtration rate, and certainly we have to look at that and see, does the mechanism of action of the compound itself alter glomerular filtration rates? And that's a key component of this, but let's not forget that it's not just the GFR that we're looking at. We're also looking at the tubular properties of these compounds as well, and that can also cause renal dysfunction, again, in a different way, but certainly a very relevant way. So we've outlined this in terms of the drug property itself. So how do we detect for this, and how do we manage that? Well, one of the key ways we do that is we look at who we're going to include in studies. And we've talked a lot about how children are often not included, or certain GFR parameters are not included. Well, that's in part because we look at the compound itself. And the compound's risk for nephrotoxicity, in addition to the clinical trial monitoring, is really what we look at throughout every development phase. And we also look at any population-based risk factors of nephrotoxicity, including the ones that I've mentioned, the concomitant therapies, the underlying disease, as well as any comorbid conditions that might exist. So how did we do this? Well, we specify inclusion and exclusion criteria. And for the purposes of renal impairment, we're often looking at exclusion criteria. And so when we're looking at this and we're trying to really identify what's going on with the compound as it relates to the patient, we want to take a very conservative approach. And in order to do that, we often do exclude patients that have impaired renal function at baseline. And we do look at serum BUN levels, we look at creatinine levels, we look at estimated GFR levels. We also, though, look at abnormal urinary constituents. We look at hematuria, we look at proteinuria, which may sometimes also affect eligibility criteria. We will often incorporate serum and urinary biomarkers. Some are validated, some are exploratory, so we really do have to provide context as we're looking at these values as well. If we see any evidence, basically, of any underlying kidney abnormalities, i.e. any congenital renal abnormalities, structural abnormalities, anything that might affect renal function, those may also qualify as exclusionary criteria. In terms of looking at the renal function holistically, we do often focus on that estimated glomerular filtration rate. And it does still hold great importance, and we do want to quantify that degree of impairment. So that still becomes very relevant and very important. And most clinical trials do have exclusionary criteria when it comes to eGFR, and you'll see that incorporated. The thresholds, however, may vary. And it varies often based on the benefit-risk profile. If this is a life-threatening condition for which this treatment could offer life-saving advantages, perhaps we can tolerate different thresholds when it comes to eGFRs, when it comes to inclusion and exclusion criteria. However, if it's a medication that would be potentially more life quality impacting, we may have different thresholds for that. So the exact GFRs that we would use for cutoffs do vary based on all of those factors, and it's important to recognize that. Despite all of what we know and how to triage and how to quantify and how to select patients, there are still a lot of mysteries, especially in the pediatric population. The eligibility in infants who have rapidly changing eGFR, which have immature kidneys, those still remain to be fully elucidated in how we include or don't include those particular patients. And I think ultimately it does come down to evaluating that benefit risk of the compound as well as the patient population. And then similarly, as we've talked about earlier as well, muscle mass, of course, makes a big difference as well, and we need to factor that in as we're using these different formula, as we're determining these eligibility criteria. Once we've established our criteria, well, then we have to talk about how are we going to actually dose these patients. And we have lots of ways that we study this. We study this through animal models. We look at on-target and off-target safety effects. In other words, are the kidneys the target? Is that just kind of the happenstance where the toxicity occurs based on pharmacokinetic properties, for which we do pharmacokinetic studies? We often will do drug-drug interaction studies. There may be dose-finding studies that have been done in animals, and then we move on to human models, including our phases of clinical development, where we look at phase 1 studies in healthy volunteers. And there are times where we do healthy volunteer studies in pediatrics, in children, so that we can establish those drug dosings. Again, it depends on the molecule. It depends on the indication as well. We sometimes may do phase 1 studies in those with renal impairment as well, to again gather that information of what do we see in terms of changes, not only in our eGFR, but also the other parameters such as tubular function that I've mentioned. We may do dose exploration studies, phase 2 studies, and then, of course, there's the whole component which we'll talk about in later sessions regarding extrapolation of this data into the pediatric population. We also know, as we've talked about this morning, that renal function is fluid, and that holds true within clinical trials as well. There may be renal events that occur that alter a patient's glomerular filtration rate, either temporarily or even permanently. And so what we've taken the approach to do is define renal events. And remember, whatever method we're using to monitor kidney function, the best thing we have here is the patient as their own control. We have the patients that we're following over time. So it's not that we're looking at one point in time, but we're looking at changes in GFR as well. And that becomes very important as we monitor the patient, as we look at their own renal function. So when we define renal events, we define them as basically abnormal clinical signs or symptoms and or laboratory abnormalities that reflect impaired function. And so when we start there, we are talking about GFR, but it doesn't stop there. We also are looking at changes in urine composition as well, and we're using our tools and the biomarkers that we have available to say what's changing, if anything. We use a lot of our clinical acumen, right? We look to investigators to evaluate these patients to say what's changed. And so we're putting this together in the context of that patient to learn more. And as we do that for each patient, we continue to learn more. Because what we know is that the patient themselves may be asymptomatic. They may or may not have symptoms that are associated with renal impairment. So we do need to follow the eGFR at various time points within the study and monitor that consistently. And given the variety of the different assays that we have, it's important to measure these patients' renal function in a similar manner throughout the study. We prefer to use a central laboratory because then we can monitor it. However, there's times where that's not practical, and we do have to look at the changes over time in these patients. And that change becomes a major determinant of the next steps in how we evaluate these patients. A lot of the safeguards incorporated in clinical trials do assess this in terms of changes that are seen in eGFR and how to manage that and how to investigate that further. For our purposes, we generally say that a 25% change in eGFR is what we consider to be significant. Having said that, that's also caveated in multiple ways and does need to be individualized. As I mentioned, that change in that GFR becomes very important. But again, it raises the question, in a child or especially in an infant whose GFR is changing dramatically physiologically, how do we incorporate these changes? And so we need to evaluate that. We really need to come together, I think, as a scientific community to tackle some of those challenges that still remain. The one thing that we can't forget, which we see too often in clinical practice, is acute kidney injury in the setting of chronic kidney disease. So keep in mind that this does happen clinically, as we've all seen, and I think we've heard a great example of vancomycin dosing in the neonatal population. Well, I think that applies with many medications, with many settings of acute kidney injury that occur. And what we have to keep in mind, just as we do clinically, is maybe we need to level set, and maybe we need to change where the patient's baseline is after an acute event occurs. And we can't keep assuming that, oh, there was recovery, we're back to where we were. There may be alterations now in pharmacokinetics and pharmacodynamics, which may affect the product and affect how the body responds to that product as well. And that may result in drug dosing changes that need to occur even within a developmental phase for this particular patient. And so how do you do that? Well, we still do rely on clinical judgment. Here's a little algorithm that we've developed in terms of how to evaluate when you do see a change in eGFR in a clinical trial and how to assess that both clinically as well as through tools using not only standard laboratory data, but sometimes even exploratory biomarkers. Then comes the very difficult decision. You've seen a change in the GFR. What do you do? Well, ultimately, it goes back to benefit risk, and that lies within the hands of the investigator and looking at the potential benefit and looking at the potential risks. We've developed some general guidelines on when study drugs should be interrupted and when it should be discontinued. This poses challenges in other ways, too, right? Because not only now have we seen the effects of drug dosing on a patient, but now if we remove them from the study, are we continuing to see the follow-up of that and to see what happens? Does their kidney function change? Does it return back to their previous baseline? Does it set a new baseline? Has it caused permanent damage, et cetera, et cetera? So it's very important to continue to follow these patients, even to resolution, whatever that resolution may look like. And as we say in clinical practice and as we teach our trainees, reassess, reassess, reassess. And I think that has been a big theme for us when following these patients, is that you want to monitor them closely for recovery. And you have to say, where do we stand now and what should change as a result of that new baseline, if there is a new baseline, in terms of renal function. And whichever formula we're talking about, it's best to, again, reassess and see where we're at. In certain situations, as we've heard, namely one of those situations being the oncology patients where small changes can make big differences, it may be important to measure the GFR rather than using any formula or estimating them. And so using that, using the tools that we have can get us to those precise measurements that we need in order to deliver drugs to these patients more effectively. There's always that opportunity, if it presents itself, to potentially interrupt the drug, reintroduce it, and look at the different properties. And we can do this through serial assays of drug level measurements. We could do that through various measurements in eGFR, and we can use our modeling techniques to really understand how drug metabolism works in children so that we can continue to learn from it. And with this, I would actually like to hand it over to my colleague, Nick Webb, who's going to give us a real-life example or two.
So, thank you very much, Deepa. Firstly, as another European, it's a real honour to be here, and thank you so much for the invitation. Secondly, I'm conscious it's me between you and coffee, so I will try and be brief, but I hope bring up some issues that might contribute to the panel discussion after the coffee break. What I want to try and do is just really outline, in a very practical way, some of the challenges faced in the recruitment of children into industry trials, children who've got reduced GFR. I'm going to start by talking about children in studies for indications for kidney disease. So these are children who have chronic kidney disease. Now, this is rare. There's probably only around 5,000 to 10,000 children with CKD in the US or Europe, and the majority of these are treated in specialist sites. This means that really any study that we do has to be on a very large scale, a global study with site-to-site referrals, and that has implications. I only moved from clinical academic practice to industry 5 years ago, so I think I can say that pediatric nephrologists certainly have been and probably still are relatively inexperienced in conducting clinical trials, and that goes the same for adult nephrologists. And it's been really important, these great collaborations between organizations like the NKF, the Kidney Health Initiative, the pediatric community, and the FDA. And with that, the development of networks like the PNRC and NAPROTIX who've really brought these studies to the fore. Now, we've heard wonderful talks this morning from people from great academic centers. And really, if we were able to conduct all of our studies in those academic centers, wouldn't that be fantastic? However, the real truth of the situation that can't happen, and that perhaps some of the standards that we hear about in these leading academic centers become more challenging, particularly in low-income resource countries. And we all know the importance of recruiting a broad base of nationalities and ethnicities for both scientific validity and also for regulatory reasons. Now, we've heard lots about creatinine measurements. There are differences in the way that you measure creatinine, and that changes the validity of the various equations that George and Guido have spoken about. And what this really means is that we have to use central laboratories, and these are really favored in industry studies. But they cause challenges. You get differences in eGFRs calculated between local and central labs, and that might change a child from being eligible to being ineligible if their GFR lies at one end of the scale. Fantastic work has been done about optimizing formulae, which is the best formula to use in which situation. The reality is, if you look at industry studies, the very large majority are still using the 2009 bedside Schwartz formula to determine eligibility in terms of GFR. We have problems with adolescents, so don't forget that things are different outside of the United States. There are 16, 17-year-olds who may be seen in adult centers, and those produce challenges. You don't get your height measured if you go as an adult to an appointment. They don't know what Tanner staging is. It's difficult. Also, this issue about adolescents moving into adult sites. So if you have a study where you're recruiting 12 to 17-year-olds who turn 18, they get transferred to an adult site. They don't get their height measured. They don't get their Tanner tested. Maybe there's a different creatinine assay. They use a different formula. You can see the challenges. When we're in kidney therapy studies, pediatric nephrologists are obviously very comfortable with abnormal GFR values. But still, the large majority of these studies for therapies for the treatment of glomerular diseases exclude patients with CKD 4 and 5 for the perception that this therapy is less likely to work, particularly if on biopsy you've got a lot of interstitial fibrosis, tubular atrophy. And there are concerns about comorbidities in this population. There are exceptions, patients with rapidly progressive GN. However, this is general practice that children with GFRs under 30 are excluded unless you're doing studies in drugs for the treatment of CKD complications, so bone disease, anemia, etc., or indeed studies of acute kidney injury therapies. However, it is possible, and this was a really nice manuscript from the FDA, were many of the authors on this manuscript, looking at the number of drugs which have been approved both in the United States and in Europe, and these were therapies principally for the complications of kidney disease, but just a marker that, yes, it is clearly possible to do well-conducted clinical trials in children with low GFR. So let's move on to clinical studies outside of the nephrology space, so studies that will be conducted when nephrologists weren't PIs. And again, I think it's fair to say that people that don't look after children with chronic kidney disease are slightly uncomfortable with the reduced GFR. They would be perhaps struggling to keep abreast of which formula to calculate GFR will be best in which situation. I think that non-kidney specialists have less understanding about normal GFR maturation, normal kidney physiology, and they'll want to have nephrology backup to help them put these children into clinical trials. And again, this impacts on choice of study sites. I'm not going to talk a lot about neonates except for the fact that they cause us huge problems. And I think that Guido has spoken elegantly about this, and I know this is something that we're going to speak about throughout the course of the meeting. The number of studies conducted in this population is really small. And then, of course, there are these challenges outside of the nephrology space with all of the disorders. So Guido has spoken about the muscular dystrophy, cerebral palsy, methylmalonic acidemia. In my last job, we had a child on the dialysis program. The highest his creatinine ever got was 60 micromole per litre. That's about 0.7 in US units. And he had kidney failure. So it's impossible to use standard formulae in this situation. And I won't replicate what Guido's nicely illustrated about children with spina bifida and in a wheelchair. These formulae are simply not validated, and it's inappropriate to use them in this population. Now, who gets excluded outside of nephrology studies? Well, certainly almost everybody with advanced CKD. It's very unusual to see a clinical trial that will allow participation in children with very low GFRs. And it's pretty common to exclude children with GFRs of less than 60. This is somewhat population dependent, so much more likely to happen in an asthma study than perhaps a heart failure study where there's a higher chance that these children will have low GFR. And then additional criteria that Deep has spoken about, so the presence of abnormalities on urinalysis, the presence of urinary tract dysfunction, or other congenital abnormalities known to impact on kidney function, and that we've heard earlier about how CACUT, congenital abnormalities, are one of the commonest causes of decreased kidney function in the pediatric population. What have I just done? Sorry. We can keep this simple or we can keep it complicated. This was a Novartis study in heart failure of sacubitril, Entresto's the commercial name. But the table on the side only shows an illustration for how, instead of just stating we wanted GFR above a certain amount, this was made incredibly complicated, looking at being above 30% of the mean GFR for that age. So a complicated table to choose to decide on exclusion from the study. I think keeping things simple is clearly beneficial in the large number of situations. What impact does excluding children with low GFR, and I've tried to find out or deduce how you can calculate what proportion of patients you will miss from your clinical trials. And this is really very, very hard to do. Though I think for most non-nephrology trials, the number of patients that won't be excluded because of low GFR is very small simply by virtue of the relative rarity of kidney failure in the pediatric population. We can ask sites to conduct screening logs, but what most sensible investigators do is actually not put children through screening who they know aren't going to automatically fail that screening process because of their GFR. So this does not give us accurate information. Oncology trials get very complicated, and we've heard Deepa and others speak nicely about this. I think that certainly where I used to work, the oncologists perhaps did more measurements of GFR than any other specialty in the hospital. These drugs often have a narrow therapeutic index, may be renally excreted, and Iahexol may have a role here. And I'm going to come to Iahexol very briefly at the end of my talk, which will be shortly. But I think pediatric nephrologists are a group that are comfortable with measuring GFR. We've heard from Guido and others about the importance of this because such a high proportion of cancer survivors have impaired kidney function. The adverse effect profile of these drugs often affects the kidney. So if you were to look up studies like the Children's Oncology Group, phase 2 combination chemotherapy, very nicely GFR determined dosing for this group. Okay, so we've heard about the CKD study because George spoke about it. This is a fantastic piece of work. I think as a pediatric nephrologist, I'd love to have been part of this study. What a fantastic, this big, huge group of children with chronic disease had regular measurement of GFR during follow-up. And this has just generated hundreds of publications and really transformed what's known about this group and improved their standard of care. The important thing, I think, is it's shown that this can be done, that GFR can be measured on a regular basis, and it's a practical thing to do. And George didn't speak about this, but I like this paper because what it did was it showed that you can actually use finger prick testing or ear prick testing for this. And this means you can do this down into neonates using heel prick testing for using measurement of GFR. I am going to take, and very respectfully, disagree with George on the statement in the abstract for the manuscript, which is where he said that measurement of GFR by Iahexol disappearance has become a gold standard in the pediatric CKD population. I think that what the CKID study has shown is it is clearly possible to do this in a range of good academic North American pediatric centers. However, the reality is this isn't used in routine practice around the world at the moment. Just to make sure in the last 5 years things hadn't changed hugely, in my preparation for this meeting, I called colleagues around Europe, and it's still relatively unusual practice outside of research settings, and it's restricted to the larger academic centers. There's some cost associated to it. It's of course invasive. It requires the 4 blood tests which George showed us in his talk. And as I said before, I think that eGFR estimation using creatinine remains the norm in industry studies. And as I said previously, the majority are still using the 2009 Fort-Schwartz formula. And I think industry, perhaps there's some reluctance to incorporate Iahexol into pediatric clinical trials until there is more widespread global adoption of it as a routine clinical measure. And these are very much my views, as Deepa said, these aren't Novartis views, but I wonder if there are special situations where in clinical trials we ought to actually be using Iahexol rather than estimated GFR. And you've heard many of these before. We've heard about using this in infants and very small children where these calculations work less well. Perhaps in oncology, where very precise GFR-based dosing may be required with highly nephrotoxic compounds. in the clinical disorders that Guido spoke about with reduced muscle mass or lower limb abnormalities. And perhaps we could even think about using IHexol for endpoints in nephrology studies. So we don't measure IHexol at every point during the study, but around the measurement of the primary endpoint, a baseline, an end of study, perhaps we should consider using IHexol in this situation, particularly where in pediatric studies in rare diseases, the duration of follow-up may be relatively small, and also the change in GFR may be relatively small, increasing the importance of accurate assessment of GFR. So in summary, and I'm summarizing Deepa's talk as well here, I think I hope that we've shown you that the drug development needs to account for renal toxicity, both due to investigational compound properties, but also the underlying disorder, that we need to be very stringent in study eligibility and how we monitor kidney function throughout the study to protect our patients, that we should assess them thoroughly using eGFR and clinical criteria to determine benefit risk. And then I hope that I've really shown you what challenges we have conducting clinical trials in a global environment, that I think that we remain generally conservative in excluding children with CKD. And I hope I've kind of suggested some ideas about where we might think about IHexol measurement, and I hope that that's something that we can pick up in the discussion after our break. So thank you very much for your patience. I'm sorry we're a couple of minutes late, but we will make up for it during the course of the morning. Thank you.
Thank you so much, Nick and Deepa. We're going to take a approximately 20-minute break, so if you can return at 11:45, and we ask the panelists and moderators for session 1 to come up to the stage at the end of the break. So please come back on time. Thank you.
The panel discussion. Welcome to the panel discussion for session 1. Thanks very much to all the speakers for your excellent presentations. We have about a little bit less than 90 minutes allotted for this Q&A, and we're going to be watching the time closely to make sure we all stick to the schedule. Why don't we start off the panel discussion with some clarifying questions from those in the room or those online for the speakers about any of their presentations.
So this question was online, came from a participant online, so it's directed towards Dr. Schwartz. Based on your presentation, how do you correct for edema and volume of distribution differences when you do a plasma clearance?
That's a very good question. We did not have a lot of severe edema in our population with CKID. This is, we were kind of discussing a little bit of what nephrotic syndrome does, and that certainly causes edema. And certainly if I were to conduct an iohexol study, I'd be very careful to get many early and late points, because the distributions are not predictable for such people. And as we also talked about, body surface area may not be the best way to normalize that GFR. That doesn't give you an answer. It gives you what you probably shouldn't do.
Maybe I can ask a follow-up question on that, and that is, it is widely used that we use the actual weight for the body surface calculation, which really leads to underestimation of the GFR in obese patient and overestimation in slender patient. And is there an approach? Is there a recommendation or so? I use the ideal weight. I match the weight from the percentiles, but isn't that something that we should all do and stop these under and overestimations?
I'm not sure I'm prepared to answer that, but I know in oncology, there often a premorbid or an early weight or an ideal weight is often used with those chemotherapeutics. But patients now, with the obesity epidemic being what it is, the ideal weight is so far away from what their actual weight is. I really don't know what is the right grounds to administer a medication or a drug.
All I can say is when you correct and then plot your GFR versus the MGFR, then the EGFR scatter is much reduced, right? And I think we need to have estimation models that have as little scatter as possible to improve the regression.
I would think that an investigation into how best to normalize GFR would be of great importance here.
Thank you both. Nikisha, are there any other questions online?
There are no other questions right now.
Okay, great. Thank you. Let's take a step back. We've really heard academic, clinical, industry, and regulatory perspectives this morning in these short presentations. And I think if we try to put it all together at a high level, when we're thinking about the best estimating equations for GFR, which equations, in your opinion, based on the evidence we've been discussing, currently provide the most accurate and precise estimate compared to true GFR in the pediatric population? Why don't we start there? And anyone on the panel can certainly chime in.
I think that if you have impaired kidney function, that currently the U25 is the best formula that is available, and one should be utilizing cystatin C and creatinine together. What is not answered as much as it should be is what do you do if you have a normal GFR or a GFR in the hyperfiltration range, because there are just not enough data points in the regression analysis. What one really should do, in my opinion, is to combine all the GFR measurements from the world, and so that you have that diversity of chromium-51, EDTA, Stockholm, I think, still doing inulin clearance, and so on, so that you have the robustness like in the CKD epi with 3 different types of GFR determination, so that that noise is eliminated, and then do a regression analysis across the entire spectrum. I know that Hans Pottel has tried to do that with the full age spectrum, but that formula, again, in the young adults is a bit underrepresented, and at least according to our data, doesn't work as well. I think we need a worldwide coordination of this and then come up with better models. But for now, if you want to have the detection of GFR over 75, that's our data, but we have to validate it externally, but with combining the modified Schwartz formula with a cystatin C-based formula.
Thank you so much. Others on the panel with your thoughts?
Maybe to add, I think, one of the challenges is not only which formula to use, but when do you switch using formulas? Especially as children get into adolescence, as they get into adulthood, and I think consistency becomes the key, is to stay with the formula and follow that patient over time. And I think that is another way we can do this. I think some of the biomarkers and using them, like as you mentioned, beta-2 microglobulin, and not that it's an absolute and it's not that you can always use that in clinical practice, but if we start looking at those and start comparing as we're doing drug development, as we're looking at dosing, and when we adjust it, that may add to our arsenal of information. And whether they be exploratory, whether they be validated, I think we have to look at all of them and come together, as you said, and maybe that's how we do validate them.
Thank you. Shamir?
I think the point I'll make in terms of that answer is the right answer today may not be the right answer tomorrow. These, as Dr. Schwartz and Dr. Phillip pointed out really well in their presentations, these formulas are constantly being adapted, improved, changed, And it behooves both the regulators and the sponsors to keep abreast of what's the most accurate formula to use at the time and make that part of their study protocols. And the lag time between the publication of these formulas and the adoption of these formulas, maybe in the greater clinical community, there is always a time lag for that to happen. So we need to be cognizant that we need to constantly be reevaluating how we're assessing GFR based on the newest, latest, and greatest updates in the formulas.
I think the evolution of the formulas via CKID do reflect the effect of puberty and sex on the estimates of GFR, and I know they will predict a different GFR for male versus female, and that's probably not physiologically correct. So there is room for improvement. But I will point out, one of the slides I did show, the creatinine-based estimates seem to do very well in a European population that had high GFR. And we think there's big room for improvement on cystatin C. And yet, I know several of the industry speakers really didn't talk as much about cystatin C as perhaps it should be, because in general, if you get an estimate with both biomarkers independently and they agree, say within 10%, then you're good. You're good because they're affected by so many different things. What affects creatinine doesn't affect cystatin C and vice versa to a great extent. So I actually look at the individuals' GFRs, and you and I know as I follow patients, for example, like some days if I got both a cystatin C and a creatinine, then the next time I get a creatinine, I'm going to get a different eGFR. So yes, you have to do everything the same way. But if you really want to know, if you get them both, you're going to be in much better shape. And as I say, the creatinine one will work better in the higher GFRs than the cystatin C one at this time. But I know CKID is planning to recruit a number of teenagers in the next iteration, and there's going to be a sub-study that's going to look at transplant donors. So we should get some new data, at least on the older kids.
Please.
I would like to add a couple of comments on that. The cystatin C has one internationally certified reference material, but I believe only 3 or 4 of the assays that are available have used that. And just like we have to have this forced standardization of IDMS traceable creatinine, we have to have the forced standardization of cystatin C and have a worldwide agreement on that, so that the worldwide the certified reference materials are used only. The issue with the sex differences, though, applies to still both, and I think that one also needs to really have an intellectual discussion if there's a way that we can standardize to extracellular volume and not the body surface area, which, by the way, body surface area is based, when you use either the Mostella or the Dubois formula, is used on data from 1906. And we have this worldwide acceleration of growth. And even Haycock is 1976. It's still 50 years of accelerated growth, and the obesity epidemic only began in the 1970s. So the whole standardization to the body surface area is a problem, because most of the height acceleration occurs in the femur and in the tibia, and the percentage rules of 9% for the front of the leg and back of the leg may no longer apply, and we may be grossly underestimating the body surface area in very tall individuals. We actually don't have anything for them. There's no reference interval for that. So the standardization which is needed to compare across the growth may have to also be revisited.
Another area that I think warrants probably a little bit more thought is not just how adequate the estimation equation is when looking at measured GFR, but measured GFR. So the question, for example, said how well does it get true GFR? But measured GFR is actually quite far from true GFR. And while we inherently know this, I think there's certain parts that might still, like I said, be underappreciated. So if you look at the P30 for most measured GFRs compared to inulin, it's 90%. If you look at the coefficient of variability for inulin just itself, an individual, it's 7%. But another part that's even less appreciated, I find, in the general community is the diurnal variation in GFR. So Koopman et al. In the Netherlands, I believe in 1989 or so, for example, did a nice study, and they had some other studies that showed this, but it was in healthy individuals. They did 24-hour inulin clearance. Morning average GFR in this group was 121. In the evening, it was 89. 30% diurnal variation, which is really fantastic when you think about it, not in a good way, of course. So if you're measuring your GFR, even if you're doing it accurately and you're doing it well, in morning and evening you get two different values. And a point in case of where this might even show the relevance of it, in the CRIC study, which is in adults, there was 1,000 people had iothalamate, very well standardized, good QC that were done with longitudinal follow-up, and they compared how it would pretty much predict both ESRD and cardiovascular events compared to eGFR, with the assumption that it would do better because now we have well-standardized measured GFR. Interestingly, the estimated GFR performed slightly better than measured GFR. Now, I can't tell you exactly why, but my assumption is that it goes back to the diurnal variation. So your serum creatinine is reaching a steady state over a couple of days, so it might be like an inaccurate hemoglobin A1C versus a more accurate fingerstick glucose. Well, one is the average versus not. But when we're considering in terms of how we want to standardize two, measured GFR is not true GFR, and unless we start thinking about diurnal variation, not an easy task, we might be missing part of the equation here.
I think that's a really valid point, but very boring practical answer. You know, when patients participate in clinical trials, the very large majority of study visits are done first thing in the morning because we collect the first morning void urine, because that's kind of when the day starts and that's when they end up in the hospital. So absolutely correct what you say, but I think the number of patients that might have, you know, their beginning of study visit at 9 o'clock in the morning and their end of study visit in the evenings, pretty small. But your point is important, that we must not consider MGFR to be a gold standard, which is completely flawless. Yeah.
I would like to add something about the quality of MGFR. So in the CKID study, were not too many patients that had severe nephrotic syndrome, but the equilibration between the intravascular and extravascular compartment is substantially delayed in those patients. And adding to this, so I did a survey amongst the Canadian centers, and most of them only use 2 time points and not 3 time points. And the first time point is too early to make sure that the equilibration between both compartments has occurred. And so the advice to the industry is not to start any sampling before 90 minutes after the injection, and to spread it out, ideally maybe up to 3 hours, which unfortunately, though, delays the measurements. But a standardization, so you cannot assume that center, even an academic center, has the best way of measuring GFR, which may explain some of the observations.
No, and we were talking about that during the coffee break, and I think that, I hope that CKID will help generate important information about this, because let's look at where the real unmet need is. You know, FSGS, steroid-resistant nephrotic syndrome, C3G, these are all disorders where there's a very high prevalence of nephrotic syndrome in the sort of disease phenotype.
To that point, we actually looked in the very beginning. We did a 9-point iohexol, and we published that in KI in 2006. And we actually had a point at 360 minutes. And for the benefit of our patients, we figured out it probably would take maybe 5 to 8 hours to get a real equilibration point, but they couldn't stay in a CRC that long. So 5 hours ended up being a compromise. So we're actually doing 2 hours and 5 hours for the ultimate study that we do now, 2-point. And if we were to go to babies, I actually talked about getting a point 2 hours and then going ahead and getting a point tomorrow. Because these are babies. Their absolute GFR is going to be maybe 5% of what the adult would be. And I think that the study is much more acute with iothalamate, but it was published by recently, and it seemed that if you truncated your study too soon, you were overestimate your GFR by as much as 10 to 20%. So yes, timing is very, very important.
And the use of two-compartmental nonlinear models, and not what most centers do is just a simple log transformation.
Thank you all for your comments. I wanted to take it back to the original question. So is there consensus that the U25 currently that relies on both creatinine and cystatin C probably is the best estimate right now, at least in patients with baseline kidney dysfunction?
I would subscribe to that if the GFR is under 75, yes.
But there still seems to be more work that we need to do in terms of the best estimate in those with normal baseline kidney function.
But yes, but that's the population you're probably least worried about, isn't it? In terms, you don't need to adjust dose. They're unlikely to be in a study of a therapy for chronic kidney disease. So academically important, but
pragmatically less so. And I wanted to take it back, Nick, to your presentation, because if we think the U25 really is indeed the most accurate, but yet we're hearing industry trials are relying predominantly on the bedside Schwartz, can you comment a little bit about why? And it's clearly it's assay dependent, but there are a lot of factors. Perhaps we could discuss those a little bit.
Yeah, I mean, I was only just reporting how things are. I wasn't. This wasn't meant to be a sort of justification of it. The reality is that the large majority of industry studies use the 2009 Schwartz bedside formula for GFR. Whether that's lack of appreciation of the important work that CKID's done and the derivation of the best formulae based on comparison of measured and estimated GFR. Maybe it's custom and practice. I don't have I'm not here to speak on behalf of the entire industry and justify it. That is how it is. I think things can be improved.
Yeah, thank you. I think, and Ash, you have a comment, please.
Yeah.
Or question.
I think it is true, modified Schwartz formula has been the one that we have used the most in the industry. But I think one of the points which is maybe more problematic is the availability of cystatin-based assays, and also being the calibrated cystatin-based assays worldwide, because our trials are worldwide, and to have the right assays available is what can cause.
So I think I was going to say that's been a big limitation, is that consistency. Plus, you're looking at the underlying state, and there's also the practical limitation, especially in children, and especially the younger they are, in how much blood we can draw off of them as well. So there are limitations pragmatically, because it's not just renal function we're measuring, right? It's also the blood counts. It's also the electrolytes. It's also. So we run into that, and then you kind of have to prioritize, what am I going to measure? And what we know, and historically what we've used, is the bedside Schwartz. So that I think we're comfortable with. It's more standardized. It's more readily available globally. The other issue that we face with some of these drug trials is the concomitant medications, namely the high-dose corticosteroids, which can affect cystatin C. And that has really put it down in terms of usability, and so we've, I think, defaulted to that bedside Schwartz. The other thing to keep in mind is a lot of these studies don't take 6 months, a year to do. They take years to do. So to change the methodology in the middle of a study becomes very difficult, or even one study to the next, a phase 2 to the phase 3 study, because then how do you put all of the data together in terms of what should the real dosing be? So I think it's because of all of those variables that we've really defaulted to the bedside Schwartz. And while U25 may emerge as superior, I do think from an applicability standpoint, it's going to take time.
Saskia, please go ahead.
Yeah, I would like to offer the way in between, because the peers without cystatin C, the serum creatinine peers formula, also so that's the newer bedside Schwartz, actually performs better when we measured iohexol in kids in the NICU and the PICU, and there were all kinds of, they were normal renal function, there was a little bit of AKI and a little bit of augmented renal clearance in these kids. We found that that formula actually performs the best, better than the old Schwartz formula. So it's not the peers with the cystatin C, but the peers only with creatinine. And as a intensivist in academia, and we are a good center, it's much harder for me to get a cystatin C than to get a serum creatinine, and to get the results within an hour. No, forget about it. I have to wait a day. So if you want to do it in a trial, it's already complicated. If you want to put it in a dose advice, it won't work, at least not for the next 20 years, I think. But that's maybe a little bit exaggerating, but I think that's the reality. So I think you have to think about a way of measuring GFR with, well, maybe with serum creatinine, but George knows better about his own formula. So,
thank you for your comment. We have some more. Lynn?
Yeah, so just a clarification. It sounds like U25, you can use creatinine, you can use cystatin C, and you can use both. And what we heard was, I think from George, that the combination of the two gives us the best estimate. But to Saskia's point, we can still use U25 with creatinine. So I'm trying to figure out, right, practically speaking, what we should be moving towards, honestly, taking into account Shamir's point that maybe something next year will be even better. But if we have something now that we know already improves our estimate in settings of renal impairment, then what are we waiting for?
Simplicity is always better, but if I were industry, and I wouldn't want to make a mistake. So in the baseline, I would do everything I could to get an accurate measurement, whether it's iohexol or both biomarkers. Then you could follow just creatinine. But the problem with the bedside is we did not have kids going into puberty. So if you're going to have long-term studies over a period of 3, 4, 5 years, and the kids are growing, the K is going to change because muscle mass is going to change. So I encourage you to use the U25, not because we published it, but because it will work better for you.
Just to piggyback on Dr. Schwartz's point, I think one of the concepts that's coming out here is to think about your study population very carefully. Are you going to be enrolling, or are you expecting patients who have limitations of using creatinine-based formulas? Are you have a spina bifida population? Do you have a population that's going to be highly represented by overweight children or adolescents, where it behooves you to think in advance about measuring a cystatin C, so you're able to adjust for these things as you go through the protocol?
George, please go ahead. Yep.
One last thing. If you're going to study, for example, sickle cell disease, you have a drug or is a gene now, a modification, you may want to get into that. These kids hyperfiltrate, okay? So in the beginning, before you get sickle cell nephropathy, their measured GFR might be 140, 160. So none of the formulas, as far as I know, work very well up there. Certainly ours doesn't, because ours was predicated on kids with CKD 1 to 3 or 4. So you have to have a different approach there.
I would echo that. We get a lot of referrals for sickle cell for chronic kidney disease, And then when you do so creatinine and cystatin C don't work, and you have to do an MGFR, and there's so many that I had a normal GFR in the end. But you can't list all of the possible elimination. You still have to have a simple approach to make it happen. And I would agree with Dr. Schwartz. At study entry, you want ideally a gold standard plus what is available in terms of, and then you can decide to pick a limited number of biomarkers for the longitudinal follow-up.
Yeah. No, I mean, that's, this is a great discussion. We do go back to Deepa's, but, we're nephrologists. We are completely focused on kidney function. There is a whole raft of other bloods that do need to be measured, and, we have a limited amount of blood that we can sample at any time. I think the availability of cystatin C and the standardization that was brought up, I think perhaps I would worry less about that, given that the large majority of studies are using central labs, and Q2 or other laboratories are available will have a single assay that they will run across all of their centres globally, and that should be standardised and covered. So perhaps less of a concern. But yeah, I mean, so the clear message seems to be, measure everything that you can to allow yourself the opportunity to pick what you think are the best formulae. I'm not sure we've got to measured versus estimate. Maybe are we coming there later? Are we?
We'll come there a little bit later. We can.
Yeah.
We'll come there a little bit later.
Yeah.
Thank you.
Because I'm interested.
I just want to add about the cystatin C assays. Turnaround time is also a major issue, and a lot of people are still using nephelometers, which gives you pipetting errors, etc. But you can actually dramatically reduce the cost of cystatin C if you put it on a platform and automate it, and that improves the turnaround time from days to 90 minutes. We get the cystatin C in one hour.
Yeah. So, I mean, the reality in a global industry study where a sample needs to be processed, leave the site, go to a central lab, get assay, it's 3 days if you're very lucky, and probably closer to 4 or 5 is the honest reality. So yeah, you do get that delay.
Well, and then a lot of times too, if it's not part and parcel and it's exploratory, it's going to get batched, so you wouldn't get that till much later anyway. And so it does raise in itself then other challenges in terms of usability.
A question for Deepa. Deepa, in the beginning of your talk, you talked about profiling different molecular characteristics to really understand the risk potential, which is intriguing. I'm just curious, now as we're getting into the clinic, is there a potential to correlate these two? It reminds me of, the 1980s, we were doing all these drug interaction studies. The labels were almost hard to interpret, where there'd be the potential to interact with one drug versus another. And then we ended up normalizing a lot of the experimental variables and coming up with a tool. Instead of the quest for one equation to rule them all, can you come up with an approach that allows you to look at a probe for understanding the generalizability across all this? This is a difficult to study population for sure, but I think that probe approach informed by an index might be useful.
So thank you for the question, and we've been working to do that, to do exactly that, is say, can we look at the characteristics of the compound, overlay the characteristics of the patient, and come up with an algorithm. And we actually just last week had that publication accepted. So we're hoping that will be coming out in the upcoming weeks. But that's exactly what we try to do, is say, based on what you know about the risks, can you stratify, and then can you stratify what you collect in a clinical trial so that you can really tailor that, and you can say, look, if it's not a high-risk compound, when I say high risk, I mean in terms of nephrotoxicity potential, that let's say it doesn't affect glomerular function, but it might tubular, well, then you can focus in on collecting X, Y, and Z versus necessary glomerular markers. So we've aimed to do that. Now, to your point, I do think it's going to take some time to validate and to say, hey, is this the best way to look at this? But I think it's at least a start to say, let's use the tools and the science that we've collected from the animals, from the PK studies that we've done, and develop an algorithm that we can implement and in clinical practice that could be used down the road, even in terms of monitoring. So we're heading in that direction, we hope.
I just wanted to make two comments in favor of cystatin C in response to some of the arguments that were made. One, I mean, if it comes actually several days later, you can have two types of things. For one of our studies, for example, we use serum creatinine on site to enroll as the enrollment criteria, and then for all our analyses and our omics and everything else, then we use cystatin C, but higher accuracy usually helps. So I'm not sure that should be counted as that much of a deterrent. And also the point that was made from the audience in terms of, you know, it might be how many years before it's more widely available, or there's a lot of push both from NIDDK, ASN, NKF to do more cystatin C, so it might not be that long, but that is still separate what is done there as opposed to starting with a gold standard. You do the best you can, and then what the community can do, some can match, some not, but I wouldn't hold what the community can do as a point there.
I just want to point out that many hospitals have taken on the cystatin C assay in part because of the independence of race, at least in terms of the measurement. It doesn't have the same racial effect that creatinine has, and that's why our hospital put it on. And it's a 24-hour turnaround, but they do it at least once a day, and it's really, I think, changed how we take care of our kids. It's worth it.
There's also a normative factor, namely that KDIGO guidelines suggest strongly to use both in adults and in pediatrics, and that forms almost a bit of a liability if you don't have it. Or having said that, in Canada there are only 3 centers that have it. So.
Hi, this question is to Dr. Schwartz maybe, and Guido as well. You mentioned that rapidly changing serum creatinine is a major limitation of serum creatinine-based equations. What would be considered stable serum creatinine, number of measurements over what period of time, especially in the context of clinical trials? If you could comment, please.
The coefficient of variation of the actual measurement of creatinine is very, very small, but creatinines do vary in individuals from day to day, and certainly that beautiful picture of what a marathon or what eating a hamburger would do is certainly very well explained by what happens with it. So getting through that, I would say it's roughly a 10% change or so, roughly. And when you have AKI, the creatinine really lags behind, and so it changes, but it's not the first thing to change. But if it is changing and it's going up, that's clearly not good. So as a clinician, you're not happy with that. And as it changes rapidly, you can't estimate, but if later on it starts to come down, that's a good sign. Not that the person's starved, but that the kidney function is recovering. And sort of as a clinician, you sort of can use that. But if you're in a study, you really not going to want that to happen. You don't want unstable creatinine. So I don't think any place would expect you to get a value today and a value tomorrow to be eligible for a study. It's more applying these equations in a clinical arena where you can get into trouble with it.
If I might add, there are 2 meta-analyses that have been published in the early 2000s about the superiority of cystatin C over creatinine for the detection of impaired GFR. And we could probably use those data because it was on something like 4,000 measurements that the meta-analysis were done. So creatinine there's a so-called creatinine blind range, right? You need only 10% to change in the cystatin C concentration to really identify impaired GFR. So they used a cutoff of 90, and you need a much higher proportion. So the area under the receiver-operated corrective plots was at least 10% lower in the meta-analysis. I would, based on that, say 10% variability of the cystatin C would be a criterion for stable kidney function. That's a fair point. Till we get it back quickly enough to know it. But I think that one really needs to advocate to have this as a standard in the institutions and to do it on multi-analyzer platforms so that you have a decent turnaround time. Because I also think that the nephelometer methods and the turbidimeter methods, because of the need for pipetting, are more prone to the sort of laboratory-induced errors.
Guido, if I could ask you a question on that. So assuming that you do get cystatin Cs and we're collecting those over time, in a clinical trial, let's say you're studying a nephrotic state. We won't even say that. And the drug works, they go into remission. So now you've got a cystatin that was done on high-dose corticosteroids, and over time, that's decreased and even potentially been discontinued. How do you interpret those results? Because they could vary.
It's funny that you asked that. So nephrotic syndrome and cystatin C is actually a little bit of a problem. So I analyzed when you spike the samples with cholesterol, because one of the problems with nephrotic syndrome is that you get this very severe secondary hyperlipidemia, and the cystatin C assay is affected by the triglycerides and is affected by the total cholesterol. There is no totally ideal parameter. But then the other thing that happens is that sometimes for, let's say, recurrent FSGS, we do LDL apheresis and give solumetrol 20 milligrams per kilo to try to get them into remission in the relapsing graft function. And that absolutely jacks up your cystatin C when you have such a high concentration. I think you still need both, or even a measured GFR in this situation. But it's well described that, especially when the child was frequently relapsing nephrotic syndrome relapses, that the estimation of the GFR is there. One could correct, like similar to the Friedreich formula, you could correct for the cholesterol and the triglycerides, but there's very little. I think I have the only publication on that, what one could do. And it was in clinical biochemistry. It wasn't in a widely read nephrology journal.
So we're going to move on to our next question. We do have several other questions we're hoping to get to in this session. Go ahead, Phil.
So we heard today about the different limitations for using GFR production equation. So, and the different key factors that may change, like our choice of the equation. So in a special population of older adolescents going into adulthood, how that changed the approach we talked about on the first question?
I think you need a list of what to use when. And some are very easy. Don't use creatinine in newborn. Don't use creatinine in a spina bifida patient. Don't use creatinine in a muscular dystrophy patient. That's easy. Basal malone acidemia, I only have 5 patients, but that's another limitation, although the literature is very scant on that. But you may end up having groups that are not so well defined and where we actually still lack the ability to estimate the GFR. I think CF is definitely, and it was mentioned also in Nick's slides, CF is definitely a group where I think you need measured GFR. Oncology is a group where you need measured GFR.
When we think about age on a spectrum, we acknowledge what you said about the neonates, certainly. I'm just curious how the panelists think about how do you identify the best equations as you're going from birth into 17? What are those choices, and how do we begin to think about it?
Also, you really want to identify AKI, right? And at the moment, our AKI definition for newborns are all based on fluid output and not on urine output, and not based on a measured biomarker. I don't have the statistical capabilities to do this, but we should be normalizing the percentiles of the GFR per post-conceptual age. And it's very important to do it per post-conceptual age and not per chronological age. And then changes in the Z-score in America, would be a very good way of identifying AKI and of identifying where were they in the percentiles and how do they change in the maturation of the kidney function. So, but you need electronic medical record systems to do these calculations for you, and you need probably a little bit of artificial intelligence to develop that, but it could be done. And that would be a much safer way, because it really seems, even though that you stop the nephrogenesis, the creatinine keeps going up in the prematurely born babies, right, because of the increased muscle mass. Cystatin C doesn't go up until they reach the 36 weeks of gestation, when you have then this hyperplasia because of the recruitment of the nephrons. And so that's why I think it's very important to do this based on post-conceptual age. If you want to do a trial in newborns on a safer antibiotic for the Staphylococcus sepsis, you would have to really think very carefully as an industry how you design this and find a way to do this mathematically.
Amir?
To follow up on that question for Guido and George, trials, inclusion criteria, exclusion criteria often have very definitive cutoffs for GFR. If you have a trial where you are enrolling patients that are less than a year of age, and you require some way to get that point estimate GFR, what do you recommend we do in those circumstances? Do we have a validated equation? In the absence of validated equation with the newer creatinine as the enzymatic assay, how do we go about getting that point estimate in that population?
Thank you. I wish I had a whiteboard because I really tried to figure this out. But if we extrapolate from Dr. Smeets, she is a doctor now, right? PhD. Okay. So 0.31 at 30 days of age, roughly, is the K, if you want a point estimate. The K at 1 year from our U25 in boys is 0.36. So 0.31 to 0.36, and you can, add 0.01 or something per month if you want. It depends how accurate you want to be, but you can get a point estimate. There's very little change in the K between 30 days and 1 year. And it's even less in the females. It's 0.33 is our 1 year K value. And there's data. I mean, we have a few kids at every one of those ages. So we feel confident about the K. We don't feel confident about in between because there's no studies and we're not allowed to do studies in healthy kids. But between 0.31 and 0.33, it's not a very big difference. And you could interpolate in any way you wanted and it would be fine.
One other thing that you could do that would be independent of these developmental changes, if you want to just establish is the kidney nephron endowment normal or not, you could look at kidney volume Z-scores, which are easy to obtain. You just need length, width, and depth of the kidney, as long as they don't have hydronephrosis, which only affects about 1% of newborns, then you can use the simple elliptoid formula for that. And Z-scores have been published by Weitzel and by I forgot the other author. There are two papers that have really looked at that with lots and lots of those data. And this would be independent for a while. I think that the hyperfiltration resulting in the significant renomegaly will probably take months before that has occurred. So especially Karen Abbott-Ball has been advocating very strongly for doing that, and it needs some more validation with biomarkers, but it is a way that you could look at it, and that would eliminate that problem.
Actually, one could say, I mean, for single, estimate, single cutoff points, I'm not even sure how much the actual value matters as much to consistency in terms of what you're starting and what other people are doing. Nothing magical happens at a GFR of 60 or 50, right? The disease started at a GFR of 120, went there, and nothing in terms of the physiology is that different at 65 versus 55. So, but consistency in terms of what's being used when it's developed, how it's measured in the outpatient clinic, inferences made from them as long as those are measured, I think it pretty much, for most cases, should cover the inaccuracies in the estimate of a single point.
Go ahead,
Just
Dr.
to add on that, we, in our U25 equations, it actually give a range of GFR. Even though it's a point estimate, there's a range. And it's roughly, again, about a 10% either way. It's 20% overall scatter. And this becomes really important because if, you're talking to a parent and the GFR is 85, it's not 90. But in fact, 90 is included in that range. And so, this in terms of recruiting children and all, it's going to affect your point estimate.
Saskia? Yeah, we've actually been struggling with this in the kids indeed less than 1 year of age. So we have used two approaches to identify which kid has AKI or not, and then base dosing. And one is to use reference values of serum creatinine. I know, again, this is all suboptimal, but it takes reference values from 0 to 1 year of age, and then you can determine the set score, and then everyone above 90 set score has AKI. Well, you can discuss about this, and then you can take different cutoffs at different ages, which are, of course, smaller time periods than you may use for all the kids or adults. So then you start to personalize it and not use one. And the other one is there's a lot of data. There's one big study using, and I always forget it's one of the radioactive measurements from a group from Belgium. They did hundreds of kids, healthy kidney in the end, where they developed normal also reference ranges for GFR using this. And you can also could use this as well, again, set scores on these data, and then say, okay, for these ages, I use this GFR as cutoff, and then measure and calculate the GFR based on the peers or the U25. Are you still with me? It's a bit complicated, but and I think nobody has really said, okay, with this GFR, we feel that it's AKI in different ages. But we have been struggling with this as well, and lots of POPI case studies where they use a GFR, absolute GFR, and they say, well, GFR determines, is a covariant in dosing. Say, okay, which GFR? Is it an absolute GFR or is it a GFR normalized for age? And that's why we've been saying you need to look at this and not just use this one cutoff. But it gets more complicated, I agree.
I'm curious if panelists have reactions to those two approaches, just because this, I think this is a pretty large knowledge gap about, this less than 1 year age group, particularly neonates. What's the best approach to begin to estimate kidney function and, accurately diagnose renal impairment?
My answer would be that it has to be age-independent Z-scores. That for the definition for the AKI, you could then use changes of the Z-score, and that would be, if it's more than 10% drop, that would be one. But I would like to point out that you can sometimes see a very significant difference what happens to cystatin C and what happens to creatinine. So in the transplant population, for instance, you may have a rise in the creatinine, and then that always triggers a biopsy, and the cystatin C hasn't changed, and then on the biopsy you find nothing. And that has more to do with how much did the child drink, right? Because that affects the creatinine. And the AKI is a very challenging situation because you can have a prerenal, and we use sort of fractional sodium excretion and so on to differentiate that, but it's not ideal. But creatinine seems to be very sensitive to that. But before you get intrinsic renal failure damage, when the cystatin C goes up, there's a discrepancy of the two. Whether or not AKI definition should be just based on biomarkers or just based on urine output, I think is a matter of debate that we haven't found the answer for.
Please, Nick.
So I'm not sure whether it's correct form for a member of the panel to ask questions, but seeing you mentioned the transplant word, Guido, I'm really interested in your opinion and George's opinion about which formula to use in this indication. You know, if we look at the number of drugs under development at the moment, the complement inhibitors, endothelin inhibitors, being developed in indications where recurrent disease happens, and there's an indication in transplantation. How well do you think these formulae work in that situation? Because that, you know, it's the bedside Schwartz formula is currently being used in clinical trials, which, you know, will hopefully lead to licensing of compounds. So it's important that, you know, if that's not right, that we decide what is right.
Well, and you use the change of creatinine as identification of possible rejection episodes, right? I totally understand that. We've been very fortunate. We had cystatin C for 17 years, and we have been measuring both all the time. I have never formally analyzed the clinical data on that, but we use both. We're very fortunate we get both in 90 minutes. And when they are discrepant, we hold off perhaps on evaluation, look at other things, looking at the Doppler, looking at whether the proteinuria has changed, looking at what the tacrolimus level has been doing, etc.
So you sort of cherry pick the result you like.
Well, but I mean, biopsy is a limited resource that you have to really think about. You can't do it with every bump in the creatinine. And my experience has been that fluid intake has a huge impact on what the creatinine is doing.
Yeah, I was always told you can't transplant the renal nerves. It's just the kidney. So you don't have the same autoregulation that the regular native kidney would have. And our kids, are told to drink 5 liters of fluids a day. It's worse than what a stone patient has to do. But somehow that does seem to keep the creatinine down. And again, I would use both biomarkers because you can't take a chance. The biopsies are not as useful as they can be. And we also look at proteinuria. We also look at drug levels, things like that.
Yeah, so just to push a little bit further, because I wasn't referring to clinical care, I was referring to, if we have change in GFR as a endpoint, most commonly a secondary endpoint in a study in recurrent disease, we've got to choose the best formula. So what would you choose?
Well, a good example are the frequently relapsing nephrotics that require a calcineurin inhibitor, right? And there is the question whether or not calcineurin inhibitors cause nephrotoxicity, but for sure, on surveillance biopsies, you see that after 2 years, you develop some stripe fibrosis, which we deem to be calcineurin inhibitor toxicity. And in a patient like that, we do follow the GFR, and if it drops significantly by more than 10%, we trigger a biopsy. In the transplant patient, it's a bit more tricky because they vary. Even when you discharge them from the hospital, in the hospital, they may have had their 2.5 liters consistently, but then they don't anymore, or they start to resume their physical activity, and always the creatinine goes up once they are being discharged. You have to use a little bit of clinical finesse in order to decide. And borderline rejections doesn't cause any need for increased immunosuppression. It doesn't make any difference.
I'll say that we've heard so far on this panel a lot of factors that really do impact how we think about, the best equation to pick. Age, certainly, availability of assay, certainly, baseline kidney function. So this question is for the clinicians on the panel. Given all of the complexities and all the factors that we have to think about, how do you make drug dosing decisions at the bedside, given all of these complexities? And I'd be interested in drug dosing decisions in the neonate, in the first year of life, and then perhaps beyond.
So in the neonate, when I have CACUT of any form, I get a cystatin C. And I've educated our neonatologists to do so because it doesn't cross the placenta. And if it's under from that thing that I showed, when it's under the 5th percentile, I have to assume that they have a reduced nephron endowment. But I always counsel the parents that I wouldn't be able to tell them for sure where they end up until they're 18 months because of the uncertainties of that, right? But we use cystatin C in that situation. It's very fortunate that we can do a microassay and we can use 100 microliters to do that. Not every lab can do that and do a microassay for neonates. But actually, you can use that microassay for any age, and you could really reduce the number of the amount of blood that have to be drawn. I was one of the inventors of this assay, so that's why I think I had enough clout to convince the hospitals. Most centers won't have that, and I think you have to develop and validate other ways. I mean, creatinine has reference intervals by age. Unfortunately, they are stepwise, and they should be really like a proper growth, like for growth, we have this all the time. So why can't we do that with laboratory markers? At the Hospital for Sick Children, they do this caliper study, and they're developing this. So that's one thing that may help. And then, what other age group were you particularly interested?
In the first year of life, beyond the neonatal period, when there's still developmental changes going on.
So I just look at that. I'm still hoping I find a student who is doing some epidemiology who can help me to do nonlinear polynomial regression on these data. But I look at the curve and then think, is it below the 5th percentile roughly or not? But that's a homegrown approach that we use amongst the 4 nephrologists in our center. For the drug dosing, there are data that if you use creatinine versus cystatin C for vancomycin dosing, that cystatin C is better. And that you can just use the absolute calculated GFR without normalizing for the vancomycin dosing, both for the individual dose is always 15 milligrams per kilo, but the dosing interval you can really estimate from that. I would be curious to hear what you do.
Yeah, the reference ranges are really for creatinine are very disappointing. You can certainly have a low eGFR and a normal creatinine, and that's so common at all ages. So we don't do that. Creatinine change from time to time is part of what we look at. We know, as I just mentioned earlier, with the K is very little different. I only learned that this week, so I would go back and try to apply that to some of my babies. But I always feel that when I ask for a drug dosing, I'll give you pharmacologists the benefit of the doubt and you know the right dose. I'll tell you the right kidney function, because I don't think that's where it's guessed right. And that's really, is the creatinine going up, is the creatinine going down? And the same thing if you can get cystatin C in that time frame, which is often not, it's often too urgent. So if I've estimated the patient comes in, the creatinine is already giving an eGFR of 60, and tomorrow the creatinine is higher, and I want to start an antibiotic, I'm going to extrapolate a little bit and say, I think the GFR is below 60, so give me a dose. And that's really what the nephrologist should do, because he's a clinician and has this capability of estimating.
Deepa, please go ahead.
Yeah, and that's just it. At our center, we don't have the luxury of getting a cystatin C back the same day. It just won't happen. So exactly that. And George, I tend to agree with you. It's the change in creatinine and monitoring that, and then looking at things like urine output. And of course, we always write. It's I think mandatory that we write in our note, avoid nephrotoxic agents, right? And so we try to do that. And I think it comes back to I'm more likely to choose an antibiotic where I can monitor levels of the drug, because I might be able to get that back, but sooner than I could get back anything else from a cystatin C standpoint. But I think, George, you're right. But the urine output, I think, can be helpful as well, especially in that neonatal population.
I mean, you can also look at urinalysis per se. Is there blood? Is there protein coming on? So that would help a little bit there. But you really are kind of uncharted waters pretty much for that.
Shamir, go ahead.
I was just going to comment that when you use reference creatinine values within the first year of life, the second piece of information you need to know is what are the normal maturational changes in the GFR. So the reference creatinine is only relative to what the GFR is supposed to be at that developmental age. And so it doesn't start at 90, it starts maybe at 50 for a 2-month-old, and where is the reference creatinine value relative to what that GFR should be. So 2 pieces of information, I think, in those situations are critically important.
But I think it's worth looking at this just from your own population. If K isn't changing very much, and you could certainly extrapolate, you could estimate these functions between 30 days and 1 year and get an idea. Because we know GFR is going to go up. It probably won't hit corrected 100 until later than that, but it's going to be going up. And I think that's a little more sensitive than reference ranges.
I think we have 2 questions in the audience. Lily, do you want to go ahead?
Sure. Maybe to expand on Mona's question, it sounds like there are multiple factors that need to be taken into consideration to understand whether there's a change in renal function in neonates and infants. But then how do we quantify the degree of renal impairment so that that can be translated into dose adjustments? How's that being done? Clinically, a lot of this data, as we heard earlier, is derived from adults. So how's that being done and managed in neonates and young infants now?
Again, there's no easy answer to this, but it's going to be the delta. If the creatinine went from 0.5 to 0.8 in 1 day, it's a lot worse than if it went from 0.5 to 0.6. And after you've seen a few hundred of these, you might say if you go up to 0.8, that's probably well under 30. This person's in shutdown. Obviously, urine output, if the patient's oliguric or anuric, that helps here. Helps a lot because it's part of the renal failure indices of both pediatric and adult rifle criteria. Those are ways to do it. And drug levels are really, I would, whatever it is, I would get my drug levels as soon as I can to see whether I guessed right or not. And it's not an ego thing, it's a safety thing.
Others on the panel have a response? Thanks, Lily. Is there another comment from the audience?
I'm going to ask that we consider the other extreme of the age group for just 1 second. It is the most common question I get from people setting up clinical trials for children. is how do we deal with patients with normal renal function or renal function that's with a GFR of greater than 75 who are transitioning from adolescent to adulthood. By far and away, this is one of the most challenging things that we deal with practically in adopting, trying to get more adolescents into adult clinical trials, where as soon as they turn 18, they start doing the I'm blanking. Thank you, CKD-EPI equation. And there's an enormous difference sometimes between the bedside Schwartz and I would assume even some of the under 25 to the EPI equation. It's practically really challenging to do this. And we're encouraging our trials to incorporate more adolescents into the adult studies, and also when we have patients who transition who are 16, 17 when they're enrolled in a pediatric study, and then they turn 18 and now we have vastly different GFRs. I understand the U25 works very well for patients who have GFR less than 75, but we have a lot of studies that are not renal studies where they have normal renal function. Curious what your thoughts would be on how we handle that population and what advice we would give to trials that are being set up or currently being run.
I can perhaps comment on what to do with your 17-year-old. We have implemented CKD-EPI for the adults based on cystatin C and creatinine combined, and we are implementing my old filler formula and the modified Schwartz formula average up to age 25, so that you can follow them longitudinally for their kidney function. But that's a single center after a single publication that has shown that approach works. And I actually really think that we need to have more data points in that population.
Okay, we've extended U25 to 25, and if your 17-year-old could be in our, at least with our formulas, successfully managed until 25. It actually goes up probably a lot more than that. There'll be a short paper that comes out with Leslie Inker, I think, is the first author, but they looked at all these formulas, and our CKD, the CKD-EPI probably outperforms, but if this is a peds nephrology patient, even they will reluctantly agree we probably should at least follow the pediatric equations, at least for a while, to see what's going on. You can't just say it's automatically 20% higher. Now, the U25 raises that K from 0.4 to about 0.5 something, so it's a 20% increase in the estimate. So we're no longer underestimating compared to CKD-EPI. It's really pretty close now. I've kind of looked at that manually. Again, the length of the study, you can use this equation. I think the safest thing would really be, I'd use both. I mean, it's just a simple computer stroke. So do the CKD-EPI, do the U25, do them all. And you can follow them sequentially and make your own conclusions.
But at the time when you introduce the CKD-EPI, your GFR goes up by 20%.
Not so much now.
Yeah. I mean, I suppose you've got to sort of think why you're doing it. And what you're principally looking at is, has this patient's GFR changed from when they entered the study to when they enter the study? And they've either been in an interventional study and gone into a long-term extension study or something like that. In that case, I mean, to me, it makes eminent sense that you continue using the formula that you chose at baseline for the duration of their participation in the clinical trial. Of course, you're going to have, at the end, you're going to have the majority of your adult patients using CKD-EPI or whatever, and a small cohort of adolescents. But then it depends, how you look at GFR. Do you do GFR as percentage change from baseline rather than a raw value? So to me, it makes sense to continue doing what you did, not to switch formula arbitrarily just because they've had their 18th birthday. And from everything else that has been said, more is more. The more parameters we measure, the greater degree of confidence we have. So that's the approach that I would take, would simply to continue that teenager with the same measurement that they had at entry into the study.
Yeah, and
adults in the study
yeah.
are going to use the CKD-EPI equation, but you are trying to incorporate down to age 12, down to age 15. But 15 and above, people seem to be more willing to use the CKD-EPI, but in that, when you start pushing it closer to 12, not so much.
Yeah.
And so if you're trying to be practical, and there's a lot of resistance to having two different equations in clinical trials. Operationally, people are very resistant to trying to have kids with one equation for measuring eGFR, and the adults being measured in a different way. I don't think it should be that way, but there is a lot of resistance to having two equations. So would you be comfortable at what level, at what age or stage would you be comfortable using the CKD-EPI equation down to? And maybe there could be some, maybe someone from the FDA could speak to how we do these studies incorporating younger and younger patients into adult trials.
Yeah, but so I won't speak for a study that I wasn't part of, but CKID has data showing how poorly CKD-EPI performs in that teenage cohort of patients. So it would be wrong to squeeze adolescent patients down a CKD line. That's clear. You're never going to persuade the adult nephrologist to adopt the Schwartz formula for their adult patients. So I think, unpopular as it may be within your organization, perhaps that might be something that you have to fight for, that there may have to be an acceptance that the teenage patients, which will always be a small cohort of the group in a lupus study or whatever, are going to use a different formula. Sorry.
No, I was going to say, and to write that into the protocol, we've done it. I mean, I don't think we've both, I think, done it. And even with, and we are as well. We're doing it at the moment, and I don't think, and again, I think the advantage is using the central lab, doing the calculation ourselves, so then the burden does fall on the company, and I don't think it's, quite frankly, a big burden to do it. So I think Nick's right, in my opinion as well, that it's a mind shift that probably just needs to happen.
We're going to have a lot of people who change providers. Not because of that, but in fact, if the study is the same, the study will use their own data, and if you started with that formula, that you would continue it, because the continuity is the key thing here, the longitudinality. So there's no problem. They could switch to an adult provider and they could start talking about CKD-EPI, but it's not going to be that much different. And the study is, for the industry, this is the way you want to do the study, with the same measurements, the same kinds of estimates.
Adding to that, the height development has finished. I mean, the amount of growth that females have completely stopped their growth, and the amount of growth that a male has is quite negligible, and maybe another centimeter until the age 22. So just looking at how does your creatinine change, how does your cystatin C change without using the GFR is another way of looking whether or not there's worsening of kidney function.
So we are just about out of time, but I'm going to let Lynn take the last comment or question.
All right, so I'm going to attempt to summarize, because I want to make sure we all heard the same thing, if that's okay. So the first thing, and the goal of this session was to say, okay, do we have a way forward in terms of understanding the central question of this session was, where is it? Right, what constitutes renal impairment and how do we measure it as it relates to drug dosing? So we did a lot more than that because we talked about, how to do baseline measurements. But I think what I heard was, number one, there is some consensus here that if we're going to be evaluating renal impairment in a pediatric population, then an estimating equation can be used. And that estimating equation for renal impairment is likely best described as using the U25 with creatinine in follow-up, but if possible, at the start of the study, to confirm it with a measurement using iohexol, and to use cystatin C and creatinine in the estimating equation to start. But then in follow-up, if we're looking at renal impairment, that the U25 with creatinine might be okay. What I also heard was that even though we're worried about, the validation less than 1 year of age, that I'm hearing now, based on the K that's been identified, as Saskia's group has described, that we have a K that can be plugged in the equation that really probably works down to 1 month, or even, maybe in that newborn age group, and that difference between the K in 1 month to 1 year is not so much that we're going to be way off. So maybe there is some extrapolation work to be done. Maybe there's still some research to be done to understand that, 1 month to 1 year. But we probably have enough to go to use that estimating equation with the appropriate K. And with the K that's now been revised for renal impairment, again, I'm not talking about normal, but for renal impairment, all the way to 25, with the increase in the K that George described, we've probably got a pretty good number to use with U25 all the way to 25 years of age. I also heard, so that's that first question, which I think is so important. The second question, or the second comment that I heard, is that whatever you start with, you should stay with. Don't change, right? And what we have to do is educate people within our organizations, FDA, industry, wherever, that it's probably not, don't be scared of using a different equation. This is one that we think will give us the best numbers or the best estimates. So stick with that. And then I think that the last issue is that, cystatin C and creatinine, may be hard on the fly when you're in the ICU with a patient. But for research purposes and for us to be trying to figure out how to, dose in renal impairment, that cystatin C and creatinine are things we probably need to start using consistently, both of them. And then finally, there's all sorts of differences for different populations, and that has to be, we have to figure out a way to do that from FDA perspective in labeling or at least in education. But from the perspective of sort of the broader population, what we want to do is make sure that we have it right for the majority of the population and understand that some of these populations will give us very different and incorrect estimations. And we have to be aware of that, as Shamir said, when we're recruiting or who's the population we're actually studying. I'll stop there. Did I miss anything big?
No, I think you got it. That was a great summary. Thank you so much, Lynn. And, Dr. Schwartz, go ahead.
Well, I think, Lynn, you had it right. I probably misspoke. If you look back at Dr. Smeetz's paper and you give me the first 5 days for creatinine, it probably works after that because the creatinine clearances were measured from birth until 30 days. So you could even use that down to 10 days of age.
Good to know. Well, I just want to thank all of our panelists for a really excellent discussion. Thank you very much.